Buenos días a todos. Muchísimas gracias por estar aquí con nosotros, celebrando esta quinta Asamblea General de la Red Nacional de Metástasis Cerebral. Para Manuel y para mí es un placer veros otra vez las caras, un año más. Y bueno, sin más dilación vamos a iniciar la jornada. Tenemos el placer de tener aquí a Marina Pollán, que es la actual directora del Instituto de Salud Carlos III, que va a inaugurar esta asamblea. Marina es una persona que es conocida por todos, tiene una larguísima trayectoria en cáncer, en epidemiología del cáncer. Ella es médico, especialista en medicina preventiva y salud pública y ha dirigido el Centro Nacional de Epidemiología, el Consorcio de Investigación Biomédica en Epidemiología y Salud Pública y ha coordinado el eje de medicina predictiva del proyecto IMPACT también, o sea que tiene una larguísima trayectoria investigadora. Y bueno, nos va a hablar como directora del ISTI en esta ocasión, desde su posición. Muchísimas gracias, Marina, por estar aquí con nosotros.
Como veis, con un papel, pero en realidad voy a improvisar. Lo que me gustaría es dar las gracias tanto a Manuel como a María Jesús por haberme invitado a estar con vosotros y me encantaría quedarme. Me han dicho que la reunión se va a grabar, así que me vais a pasar el link porque lo de venir a inaugurar y no quedarte, pues al final es como un poco pobre porque yo no aprendo nada. Quería daros la enhorabuena por haber consolidado esta red de trabajo, que es una red multidisciplinar, que según tengo entendido comparte investigación básica con tecnología y con los servicios de 21 hospitales. Por experiencia sé que la colaboración no es fácil. Cuando venía para acá me estaba acordando de cuando empezó el ciber, salió un artículo, creo que fue en Nature, ya no recuerdo muy bien, que hablaba de la dificultad de colaboración y tenía una imagen, un cartoon de estos, un dibujito, de dos clínicos que se estaban dando la mano, no sé qué, y por detrás se estaban poniendo los cuernos. Y a veces colaborar es compartir, colaborar entre especialidades que no son la misma, también significa desarrollar un lenguaje común. Yo he trabajado con biólogos y al principio ellos hablaban de tendencia, y para los epidemiólogos una tendencia es una relación dosis-respuesta, y para los biólogos, no sé si sigue siendo así, pero era una P que se acerca a 0,05 y que al principio una misma palabra significa cosas distintas. Y cuando trabajaba con genetistas, ellos hablan de penetrancia, y lo que ellos llaman penetrancia, nosotros le llamamos incidencia acumulada. Quiero decir, con dos ejemplos muy simples, ¿no? de mi propia experiencia colaborativa, pues manifiesto el éxito de vuestra red, que es capaz de aunar conocimiento y lenguajes que a veces pueden ser diferentes, ¿no? Así que quiero daros la enhorabuena por eso, por haber sido capaces de colaborar y compartir, de pasar del yo al nosotros, que no siempre es fácil, y del mío al nuestro. Y también daros la enhorabuena por el tema de la red, las metástasis. Yo que he estado trabajando mucho tiempo con clínicos en epidemiología del cáncer de mama, la otra cara de la moneda del cáncer de mama son precisamente las mujeres con metástasis, ¿no? Las que no van bien, y a veces en tumores que tienen gran supervivencia es una historia de éxito, pero a veces nos olvidamos de precisamente aquellos pacientes que desgraciadamente, bien como decía mi jefe de Johns Hopkins, que era colombiano, aunque trabajaba allí, hablaba del esperamiento, sería de la tardanza. A veces es la tardanza del sistema de salud en diagnosticar y otras veces es la agresividad tumoral, pero el hecho es que tienen una perspectiva muy diferente y creo que vuestra red se dedica a la parte difícil, que es muy necesario investigar y avanzar, ¿no? Y también os doy la enhorabuena por el nombre. Es un nombre precioso, RENACER. Me imagino que no sé hasta qué punto tenéis relación o colaboración con asociaciones de pacientes, pero creo que a las propias asociaciones les gustará mucho ver un nombre como este, ¿no? Así que pues os doy la enhorabuena por todo esto. Creo que haber constituido y consolidado una red como RENACER os sitúa en una posición excelente para conseguir y tener, y seguro que ya lo tenéis, más éxito internacional. España, en aquellas partenariados, en aquellas convocatorias que nosotros cofinanciamos, cáncer siempre es una convocatoria de éxito, es decir, el retorno que consigue España es bastante superior a los fondos que desde el Instituto de Salud Carlos III ponemos para cofinanciar las convocatorias. Y de hecho estamos presionando para que Transcan tenga una continuidad. En Misión Cáncer somos importantes, como sabéis, como país hablo, ¿eh? Cuando digo somos no penséis que es el plural majestático, hablo de todos. Así que os doy la enhorabuena, el compromiso en la medida de lo posible del Instituto de Salud Carlos III de ayudar a consolidar cuando podamos, cuando tengamos fondos, pues estructuras tan necesarias como esta, pero en este momento estamos en unos momentos que no son fáciles y bueno, el hecho de que esta sea la quinta reunión, si no estoy confundida, pues muestra esa consolidación de vuestra red, esa capacidad que habéis logrado de poder avanzar juntos, de llegar hasta el paciente, de no quedarse en el animal de experimentación, que es muy útil, pero que muchas veces no refleja totalmente lo que es necesario para acabar cambiando y mejorando la vida de los pacientes. La misión del Instituto de Salud Carlos III es mejorar la salud de la población a través de la ciencia y vosotros nos estáis ayudando a hacer esto con vuestra línea de investigación. Así que muchísimas gracias, espero que la reunión sea muy fructífera y de verdad que en mi casa la veré. Un fuerte abrazo a todos.
Bueno, muchas gracias, Marina, por tu intervención. Desde la coordinación de la red vamos a presentar, como todos los años, cuál ha sido el avance en los últimos meses, en el último año de la red. ¿Y dónde estamos ahora? Voy a empezar yo contándoos un poquito cómo está la red, la cohorte, y después Manuel os contará más a nivel de investigación cuáles son los últimos resultados. La red nacional la conocéis todos, creo que prácticamente todos, pero la pongo aquí para por si hay alguien que se haya incorporado últimamente. Tenemos las nuevas incorporaciones, ya las sabéis, el Hospital de Santiago de Compostela, que ya ha comenzado a incorporar casos, y el Hospital del Mar, que está por iniciar, pero ya con todo listo y preparado. 17 hospitales operativos, todos ellos participan de una manera muy activa, participáis de una manera muy activa y os damos las gracias por esto, porque hacéis, lo sabemos, un esfuerzo enorme en reclutar pacientes, tanto para la colección de cirugía como la de biopsia líquida, y eso hace que año tras año vayamos incrementando el número de donaciones de las dos colecciones. Evidentemente 2025 aún no está completo, nos falta el último trimestre del año, pero por la tendencia que llevamos vamos a aumentar incluso los valores del año pasado. Quería aprovechar este año para haceros un poco un descriptivo de la colección que tenemos, de la cohorte, que yo creo que hasta ahora no lo hemos hecho exactamente así. Tenemos ahora mismo 566 pacientes dentro de la cohorte, de los que hemos recibido 1.252 muestras, un número muy importante ya, y esto en procesamientos, alicuotados, han generado más de 15.800 muestras, con lo cual son unos números ya con muchísima relevancia. Es una cohorte en la que los pacientes tienen una edad media de 63 años, está más o menos pareada, y sabéis que recogemos muestras de cirugía. Tenemos ya 407 pacientes, y de biopsia líquida, de los cuales tenemos 190, algunos de ellos en estudio longitudinal. Tenemos ya 100 pacientes en estudio longitudinal, de los cuales 8 ya han hecho los 2 años de seguimiento que tenemos un poco pautados, y 50 aún están en seguimiento activo. A nivel de tumores primarios, como sabéis, la colección de biopsia líquida se centra exclusivamente en tumores metastásicos procedentes de primarios de pulmón, mama y melanoma, pero en la subcohorte de cirugía tenemos una enorme variabilidad de tumores primarios y esto enriquece mucho la colección porque nos permite abordar diferentes cuestiones. El valor añadido que tiene nuestra colección, además de las muestras biológicas, es la información, esta completísima información clínica que nos ayudáis día tras día a recoger. La base de datos ya sabéis que se renovó por completo al inicio de este año y poco a poco hemos ido incorporando aspectos nuevos. Para eso están trabajando muy activamente nuestra Clinical Data Manager Carmen Ortega y la unidad de bioinformática del CENIO, con nuestros dos asesores externos, Ariane Stendel y Eduardo Isidor, que están haciendo un trabajo muy enriquecedor para nosotros. Hemos incorporado hábitos tóxicos, hemos incorporado imagen médica, estamos incorporando variables correspondientes al estudio neurocognitivo, estamos perfilando muy bien las ontologías, los diagnósticos, las variables moleculares y perfilando también los tratamientos que son propios de la metástasis cerebral, definiendo qué metástasis se ve afectada por cada tratamiento para que luego los análisis posteriores sean mucho más detallados. También los datos asociados a las muestras, toda la caracterización que estamos haciendo. Tenemos un reto con esta información tan completa, lógicamente, que es la actualización de la misma. Tenemos un nivel de complejidad muy grande y tenemos todavía muchos campos que no están completos, no todos van a estar completos, pero tenemos también la idea de que algunos pacientes pues, no tenemos un seguimiento muy actualizado. No obstante, somos conscientes de que tenemos casi la mitad de los pacientes vivos, con lo cual tenemos que seguir actualizando información y agradecemos de verdad el esfuerzo que sabemos positivamente que hacéis todos y cada uno de los centros colaboradores por facilitarnos esta información a pesar de la carga asistencial que tenéis y esto para nosotros es como un regalo para nosotros, que nos dediquéis este tiempo y esta dedicación. No obstante, estamos intentando buscar alternativas para evitaros esa sobrecarga de trabajo, estamos explorando la posibilidad de conexiones vía VPN desde la Clinical Data Manager con los hospitales, estamos ya haciendo estamos explorándolo con alguno de los centros, si esto vemos que sirve, trataríamos de expandirlo si hay posibilidad a otros centros para facilitaros y aligeraros un poco esta carga. También hemos visto en los últimos meses que es muy útil que a la notificación de los casos nos enviéis la información lo más completa posible, porque eso lo que hace es que la revisión de los casos sea mucho más rápida, porque vamos a actualizar solamente el seguimiento. Entonces, esto ya lo hemos testado con varios hospitales y os instamos desde aquí a que nos ayudéis en este sentido cuando nos enviéis los casos. A nivel de caracterización de los pacientes, tenemos caracterización ómica, la conocéis y os hablará luego Manuel extensamente de ella. La caracterización ómica está hecha en un porcentaje altísimo de los casos. Estamos iniciando el estudio neurocognitivo, es el que está un poquito más retrasado, y también estamos trabajando junto a MOLAB, al Laboratorio de Oncología Matemática de Castilla-La Mancha, con la imagen médica. Voy a hablaros un poquito del estudio neurocognitivo, que como os digo es el que está un poquito más retrasado. Sabéis que tenemos dos aproximaciones, una aproximación mediante evaluación más clásica, más aprobada en uno de los centros, que tenemos la suerte, la fortuna de contar con una psicóloga que está viendo los pacientes de metástasis cerebral. Tenemos ya 10 pacientes evaluados, con algunos de ellos evaluación pre y post cirugía. Y luego, por otra parte, estamos en la implementación de este Amsterdam Cognition Scan, desarrollado desde Ámsterdam, que se ha ido implementando en varias fases de piloto. Tenemos ahora mismo 6 pacientes a los que se les ha dado acceso al test. Es verdad que nos estamos encontrando una respuesta un poquito parcial. Solamente uno de estos pacientes ha terminado de completar el test, el resto o no lo ha hecho o no lo ha terminado. Por eso creo que es bueno aprovechar esta oportunidad para hacer un repaso de cuál es la importancia y cuál es el procedimiento para este test. La importancia de este test es dar la posibilidad de evaluar diferentes habilidades cognitivas que son muy parecidas a las que se evalúan mediante aproximaciones clínicas en aquellos hospitales que tienen un psicólogo dedicado a esto, y nosotros, juntando esta información de la evaluación neurocognitiva con los síntomas neurológicos que se reportan al diagnóstico de la enfermedad, se puede hacer una evaluación de cuál es el impacto que tiene la enfermedad, el tratamiento de la enfermedad en estos pacientes. Y es un dato que nos parece muy relevante por su impacto en la calidad de vida. En cuanto al procedimiento, sabéis que los que ya estáis enrolando pacientes para el estudio neurocognitivo, sabéis que tenemos un consentimiento específico para estos pacientes, que es específico porque incluye esta necesidad de que ellos hagan el esfuerzo de rellenar el test. Se notifica el caso al biobanco. Nosotros directamente lo registramos en el sistema ACS y generamos el link para enviárselo al paciente, que puede realizar el test bien en el hospital o bien en su propio domicilio, si tiene posibilidad, y esa información desde el biobanco accedemos directamente a ella para incorporarla en nuestras bases de datos. Es muy importante que los hospitales que estáis enrolando casos para el estudio neurocognitivo intentéis priorizar el reclutamiento, porque lógicamente vosotros veis a los pacientes, sabéis si el paciente tiene alguna característica clínica que haga difícil que realice el test, pero es un recurso muy válido, es un recurso que no solamente, nos puede dar una idea de la calidad de vida que puede llegar a tener ese paciente y, por tanto, os pedimos que intentéis priorizar el reclutamiento. Estamos también registrando cuando los pacientes no pueden realizar este test, por qué no pueden realizar este test. De alguna manera, eso nos ayuda a ver cuánta viabilidad vamos a tener en la implantación en la corte completa. Es importante recordar que es imprescindible que los test se realicen antes del tratamiento intracraneal, ya sea cirugía o radioterapia. Si se realizan después, los resultados desgraciadamente no van a ser válidos. Y como digo, que firmen estos pacientes el consentimiento dedicado a ello. Aprovecho para mencionar que los consentimientos informados, que son unos documentos bastante dinámicos, se van adaptando a las necesidades del objeto para el cual se recogen, van a cambiar en breve, en las próximas semanas. Esperamos enviaros una versión nueva de los consentimientos informados que nos han pedido desde el Delegado de Protección de Datos de nuestro centro y también desde el CEI del Instituto de Salud Carlos III, para adaptarlo a la nueva dinámica de compartición de datos que estaba un poco regular, registrada. Por último, os quiero comentar muy brevemente los análisis que están llevando a cabo desde el Laboratorio de Matemática Oncológica en relación a los análisis de imagen. Este es el flujo de trabajo que está establecido. Las imágenes se coleccionan, se anonimizan, se identifican las lesiones y después se segmentan para extraer de ahí aquellos parámetros que son interesantes para un estudio posterior. Se está realizando este esfuerzo de análisis de imagen tanto en pacientes de cirugía como en pacientes de la corte de biopsia líquida. En los pacientes de cirugía no me voy a entretener mucho, Manuel os va a comentar mucho sobre esto, pero ya hemos iniciado el análisis de las imágenes para definir parámetros radiómicos en relación con los clústeres terapéuticos que están definidos. Y en el estudio de radioresistencia, las imágenes están más analizadas, está más avanzado el estudio de las imágenes, tenemos ya coleccionados 102 pacientes y se han segmentado 136 lesiones, y esto nos permite ver cuál es la evolución de cada una de las lesiones, como veis en el gráfico, a nivel de imagen en estos pacientes. Por último, comentaros que para intentar agilizar el flujo de trabajo y como parte del trabajo de MOLAB, se están testando, se están buscando y testando algoritmos de inteligencia artificial para agilizar las segmentaciones, que es un proceso que ahora se hace de una manera muy manual y lleva muchísimo tiempo, y para intentar que esto vaya un poquito más rápido, están trabajando en este sentido también. Y con esto os he dado un repaso de cómo está la corte y le doy la palabra a Manuel para que revise la información más científica.
Gracias, María G. Voy a cambiar a inglés las diapositivas van a estar en castellano, yo creo que todo el mundo va a poder seguir y así facilitamos que nuestra invitada Nuria pueda seguir también la presentación. So I'm going to give you an overview of what are the more recent publications. Basically, we had one publication already there, which is about how to improve immunotherapy for patients with brain metastases. It was published this year in a high-impact journal. Basically, the take-home message is that we have a new strategy to improve immunotherapy for patients with brain metastases. We found this by combining immune checkpoint blockade with an STAT3 inhibitor called silivinin. You can see that there are these 4 aims of the experiment. On the right, you have the improved benefit by the combination of this inhibitor of local immunotherapy and immune checkpoint blockade, where that it allows us to have more CD8 T cells infiltrating within the metastases that will be killing more cancer cells. We are very lucky that our partners in Turin are running this phase 2 randomized multicenter trial with this STAT3 inhibitor, and we think that we can maybe get to the combination with immune checkpoint blockade. And interestingly, very recently, our long-term collaborator, Joaquín Bosch Barrera, has been reporting a case study in which they have found that indeed a combination of immune checkpoint blockade with silivinin, with this STAT3 inhibitor, might get improved benefits for patients, at least in this case report. This is just some images in which you can see that after 5 months on this STAT3 inhibitor with pembrolizumab, the metastatic lesions are really responding in this patient. And this is very good news and give us a lot of optimism that we might get to do this second trial if the first one is positive. Now, the other main finding of this year that is now available in bioRxiv and is in revision is that we have found an additional strategy to treat brain metastases. Here you can see our results in experimental models. This is a compound called ibudilast, which decreases very significantly brain metastases. This compound is targeting not the cancer cells, but a component of the microenvironment. In particular, these microglia macrophages that are characterized by the presence of this molecule here, this receptor, that basically help the cancer cell to grow. And what we do with RENACER is really to go to the next step. We do you can call it an ex vivo clinical trial. So we find this molecule that is effective in experimental models, and then we get access to these patient-derived organotypic cultures, 26 organotypic cultures, 26 patients, different primary sources of brain metastasis. You can see that with this compound, ibudilast, we have benefits in most of the patients, as you can see here. All these patients are reducing very significantly the viability of the cancer cells. But it is true that we can also get to the differentiation between those that are responding and also those that are not responding. And I can tell you that with this profiling that we are doing in all these human brain metastases, we identify a signature of 11 genes that allow us to differentiate the patients that are going to be responders and the patients that are not going to be responding to this therapy. Interestingly, this creates signatures of poor prognosis. So we can see whether the patient is going to have high levels of this signature, meaning that this patient will be poor prognosis. We can also identify biomarkers to stratify patients in the case that this moves forward to a clinical trial. So we as you can see here, with cerebrospinal fluid, we can measure the levels of this molecule, which is the ligand to the receptor, and we can see that in the majority of the patients with brain metastasis, we see huge levels of this molecule, suggesting that in the future we might be able to give the therapy together with the initial biomarker. So these are the things that are completed, but we have many other things that are in the process. I just want to mention a couple of things. We have a project on prevention of metastasis that I'm pretty sure that we will be showing you next year. But I want to just spend a little bit more time on this effort on characterizing 300 human brain metastases and how this is moving forward in the RENACER network. Specifically, I want to mention that all this data that we are creating and generating in RENACER, our main intention is to make this public, to help as many researchers as possible. And very soon, hopefully within next year, we will make public this RENACER CBio portal that everyone will have access. Everyone will be able to analyze these 300 human brain metastases with transcriptome profiling and whole exome sequencing. And this is a work that has been done by the bioinformatics unit at CNIO, led by Fatima Al-Sarur. We not only dedicate our time and efforts to basic translational projects, but also clinical projects. Here you can see 3 ongoing clinical projects led by Dr. Pérez, Hospital 12 de Octubre, Sepúlveda, Hospital 12 de Octubre, and Dr. Pellerino, who is in an associated hospital, Hospital de Turino. And basically you can see that we have different projects. One is a biomarker for radioresistance. Another one is a compound that radiosensitizes cancer cells. And the third one is the one that I just mentioned, a strategy to decrease local immunosuppression. Just to give you a very brief highlight, so we have, as Maria G. mentioned, already 190 patients recruited for the first study, with 98 included in longitudinal imaging follow-up, which are very high numbers. The second project is going very well. Basically, is initially developing glioblastoma as a proof of concept. In mid-2026, we will have the first data made public. This has been presented both in ASCO and ESMO. And the idea is, of course, go to brain metastasis, because the same mechanism also happens in brain metastasis. And hopefully, we will be developing a clinical trial dedicated to triple-negative breast cancer patients with brain metastasis. And the final one is an ongoing clinical trial in which this drug is provided to patients after neurosurgery. There are 17 patients randomized in the protocol, and we are developing also efforts on implementing the clinical trial by developing patient-derived organotypic cultures that we treat here in Spain in the laboratory, trying to add more value to these organotypic cultures to inform clinical trials much earlier than the results are seen by the patients and the clinicians. So, of course, RENACER is self-sustained in the sense that we are depending on ourselves to have funding to sustain this and to do the many things that you are seeing. And I have to say that we have good news. This year we have attracted a couple of projects, one from the Asociación Española contra el Cáncer and another one from a pharmaceutical company, which is also very interesting because that means that the pharma industry are seeing that they can partner with us and we can, of course, make efforts together towards getting faster to potential approvals or use of drugs in brain metastases. This is an ongoing effort. Many of our time goes to ask for money, the more, the best, of course, and as you can see, there are different strategies that are ongoing. Not only the, let's say, classical applications for funding agencies, as you can see there, but also we are starting to see the potential benefits for the network to move results that we have into the commercial scenario, and we are applying to different funding agencies, including the ministry. We are also playing at the European level, so as you can see here, we are applying to this big European consortia led by... All right, it's okay. So we, and finally, what we are trying to do also is to exploit philanthropy. This is something that CNIO is being implemented with renewed efforts, and we are trying to see whether we can specifically attract more people to help clinicians to get those data, so more clinical data managers. I just want to mention that given that we are very happy the way RENACER is developing, we get more and more ambitious, and we are not only focused on Spain. Of course, we want to attract more hospitals, but we are already thinking about the European level, and we recently submitted an application to create a European network of brain metastases, in which many friends and colleagues throughout all over Europe, if we get funded, of course, we will be invited to join, and this is something that is very attractive and will be very beneficial. So usually we also identify things that we have to complete, and these are the things that we have been showing in the last meetings, General Assemblies. We take this seriously, so when we say that we are going to pay attention to all these things, we do. So I just want to mention that we have been able to put this green light on several aspects that are very familiar to you, so you need more help. And we have a fantastic clinical data manager, Carmen Ortega, who is really transforming the way that we are working in RENACER. We have developing many things that María already alluded to. I just want to maybe focus on the things that we have been developing efforts this year. I told you already about this data portal that is really a reality. It's in beta testing now, but we'll be making public this soon. And I want to touch on this strategy to revert benefits for those clinicians that are participating in RENACER. I think this is very important. So it's not always that we are asking for things, but we should also be able to give it back. No? And in that sense, we have been thinking on how to do that. And Fatima and her team has been trying to develop a report on the specific samples that are submitted by the clinicians to RENACER. And we now have implemented a data report on the different profiling of this human brain metastasis that will be otherwise not available, of course. We are trying to define what is the content of that. For instance, on the right side, you see that we can also provide possibilities to have drugs that will be effective against this brain metastasis, at least using a predicting algorithm. But yeah, I think this could be one of the new efforts that we might be implementing that might be useful for clinicians. The other aspect that it was already alluded to is this cognitive evaluation. I think this is very important. We really want to take into account not only the possibilities to treat patients, but also to improve their quality of life. This, as you know, is more and more important, included in many clinical trials. Implementation is tough, so we have been trying to do that with different strategies mentioned by María, and we are trying to be as innovative as possible. And it's clear that there is need to implement new systems, new technologies. And in that sense, we are starting to think about the possibility of implementing artificial intelligence in the clinic, in the sense that there are emerging companies that are really aware of the problems of getting this data, the interaction with the patient, whether it does at home, whether it does it at the hospital, the time it takes to do the test, and how to track all this data and to transfer to databases are things that are already in the pipeline of different innovative companies, and we are starting to interact with them to see whether we can improve this, because we are really committed to generate this data, to take into account the quality of life of patients. Finally, I just want to mention an ongoing effort, which is really thanks to the work of people in the network that basically are paying a lot of attention to develop different aspects. Pilar Caro, mainly, of course, to develop different strategies to help patients to be informed about what we are doing in RENACER. For instance, we have a dedicated website. We have a lot of efforts to put in social media. We have created videos that patients, that clinicians or researchers can visualize. Importantly, we have also been able to attract the attention of the media, and the most important thing that we have seen this year is a full report in El Mundo, a very well-known newspaper in Spain, that dedicated a full report to RENACER. And I think this is not only good for RENACER, but I think it's good in general for helping the society to get to know why these donations are used for in cancer. I think this is very important because the article goes from the operating room to the laboratory to the biobank and the uses that we give to these samples. Something that I want to mention, as you can see, Pilar is investing a lot of effort on this. So we have been in contact with many societies, clinical societies, patients association. We are very grateful for the support of GEPAC. trying to help each other, to disseminate our initiatives, to disseminate our results, to help them to, with their own conference, to establish long-term interactions. And this is ongoing, and we have managed to establish interaction with GEINO, with EACR, with GEPAC, Asociación Española contra el Cáncer, and I'm sure many more will follow. And finally, of course, this is an enormous effort. You don't realize until you start thinking about the many people that are involved in here. And interestingly, this is a very nice equilibrium that makes this very innovative network to work, from patients that are donating their samples, to patient advocates, to the members of the biobank, the bioinformatics unit, all the labs that are involved, of course, the clinicians that are developing efforts to send us samples, and to take some of their time to participate and to make this effort possible. And I think that's basically it. We usually, after each talk, we open the turn for Q&A. So if anybody has a question for Marieje or for me, please feel free, maybe to what we have discussed or other things that are related to the network. And yeah, thank you very much for your attention. ¿Hay alguna pregunta? Is there any question related to what we have discussed so far? Yes, Ernest.
Thank you for the summary that I am not in the network, but will be happy to join. But I have a question that could be a barrier for some centers. Are you considering a federated biobank instead of concentrating to everything? Because for some centers that we have been collecting samples, it could be a bit more difficult to ship all the samples to Madrid, but maybe it will be nice to consider a more open-mind approach that the samples could be retained in the different partners, and that could be even more important when we move to Europe.
Yeah.
Yeah. Well, in fact, this is something that was considered at the beginning of the network, but as the experiments with the PDocs and so on need to be done in a short period of time, and the quality of the samples is quite important for all this study, we decided to move from this federated biobank to a centralized one. So these are the reasons for this.
So I think there are part of the things that we are doing that is not possible. At least nowadays, we have been thinking about how to do these PDocs. If we are not centralizing this might be very challenging. But maybe in the future we are evolving towards this situation, because of course, I mean, we are vertebrating the efforts in Spain, but it's true that not every region in Spain is represented. So we have a lot of work now. If every hospital in Spain will be involved in RENACER, that will be a completely different scale. At the European level, for instance, this is something that we have to do. We cannot just centralize this in a single institution. So in this European network, this will be the way to go, definitely. Within Spain, we will see. At the moment, we are super lucky that the biobank is working very, very, very efficiently and making this possible. Yeah. ¿Alguna otra pregunta? Any other question? Yeah, go ahead.
Thank you. It is really impressive what you have achieved in few years. I have a clinician question, because I was impressed by these 3 clinical trials you already have as part of the RENACER network, and we are facing a lot of difficulties to fund academic trials. So I was wondering if it was easy, if those are academic trials or sponsored by pharma, and if academic, if it's easy in Spain to have IIS trials in that setting.
I think we have limited experience, as you see. I'm not a clinician, sorry. So basically, the Aceleragon trial is sponsored by a company, the company that has Aceleragon. And then the other trial with silibinin, it's even more complex because there was the clinician involved there managed to initiate a company that is producing the drug. So it didn't happen immediately. Let's say it's not so easy. No, it's not. So far, no. But our experience is very limited, I would say. Yeah. Any other question? Okay. So now it's my pleasure to welcome Nuria Koteki. Nuria Koteki is a medical oncologist in the Department of Medicine, chef de clinic in the medical oncology clinic, medical lead for early drug development at the Institut Jules Bordet in Brussels, Belgium, and also the executive officer for the OncoDistrict Network, which is a network dedicated to improve clinical trials, generate clinical trials. Nuria has been a reference person for me because she has been crucial to develop one of the very first efforts to have a dedicated network for brain metastasis, which is Brainstorm. And as you will see, we will learn about this pioneer effort. So, Nuria, thank you very much for coming. It's really a pleasure to have you here.
Thank you, Manuel, for inviting me to present today the Brainstorm program. So as I mentioned, it was created, or the idea came in 2018, but the program was only initiated in 2020, and I was really impressed to see how RENACER evolves rapidly, and we are facing also several hurdles in the development of the platform. We can discuss that later on. So please, you take a look at to my outline. So apologies because I know every one of you is aware of what is happening in the field of CNS metastasis, but it felt a bit difficult for me to introduce the platform without introducing also the background and the rationale of why we have developed this Brainstorm program. So I'm gonna quickly talk about that before introducing the Brainstorm program. So you know that brain mets are common, more commonly seen in lung cancer, breast cancer, and melanoma, and that incidence is increasing due to both improved diagnostic techniques and prolonged survival thanks to recent systemic therapies. Recent advances in the management of CNS metastasis are based on improved radiotherapy and surgery techniques, but also recent demonstration of efficacy of targeted, including ADCs and IO systemic therapies for the treatment of brain metastasis. And this allows an increased multidisciplinarity in the management of CNS metastasis. Here you can see on the slide the most recent systemic treatments who have showed intracranial efficacy in breast cancer, lung cancer, and melanoma. And I have chosen two particular examples that seems very important to me. The very well-known, I suppose, trastuzumab deruxtecan that has consistently demonstrated higher intracranial response rate as compared to other available treatments in HER2-positive breast cancer and more recently also in HER2-low, and including leptomeningeal disease, and lorlatinib, which is TKI, that next-generation TKI that is used in ALK-positive non-small cell lung cancer, and that have demonstrated, interestingly, an improved PFS, but also reduced CNS progression in advanced ALK-positive non-small cell lung cancer. And you can see here that, if it works, correct, no? That for patients with baseline brain metastasis, the 5-year outcome from the phase 2/3 CROWN study shows that the probability of being free of intracranial progression was nearly 83% with this novel TKI. And more interestingly, the probability for patients without baseline brain mets to prevent development of brain mets was 96% with lorlatinib as compared with the previous treatment, crizotinib. But we are still, all of us, or clinicians, seeing patients that are not doing well with CNS metastasis. And I have chosen the example of this patient, one patient of mine, Valentine. She was diagnosed in November 2024 with de novo PD-L1 positive metastatic triple-negative breast cancer with asymptomatic CNS involvement from the start. She undergo first-line carboplatin gemcitabine pembrolizumab, but unfortunately no intracranial response. And as the disease was too extended, especially leptomeningeal involvement, stereotactic radiotherapy was not possible, and we had to start whole brain radiotherapy and second-line Trodelvy for this patient. We managed to get intracranial response, but unfortunately it did not last very long. And the patient had, in September 2025, a symptomatic development of multiple new brain and leptomeningeal enhancing lesions compared to June 2025, and no further local treatment possible. So she started third-line systemic therapy, but with very low expected CNS response rates. This is underlining that CNS metastasis remains a high unmet need in many cancer subtypes. On top of the limited therapeutic options, several other challenges had been identified before we initiated Brainstorm. Lack of predictive biomarker for CNS metastasis and response to treatments, lack of prevention strategies, quality of life and neurocognitions, as you have also very well mentioned, that remain often impaired in patients with brain mets, unresolved questions on the blood tumor barrier, and biological heterogeneity of CNS metastasis that remains a challenge for targeted treatment in this area. So there are various approaches to tackle the challenge of CNS metastasis that includes improved knowledge on epidemiology of brain metastasis, better understand biology of CNS metastasis to be able to rethink clinical trials for CNS metastasis, and this should be done in a multidisciplinary manner, both on the clinical path, but also in the research pathway, from basic research to translational research and clinical research. And that is what Brainstorm tried to do when it was initiated. Brainstorm has been initiated in 2020. It's a multi-centric interventional study that is sponsored by Jules Bordet Institute under the umbrella of the OncoDistinct network. The study or the program is divided in 3 parts. Part A, before the diagnosis of brain metastasis, Part B, at diagnosis of brain metastasis, and Part C, after the diagnosis of brain metastasis. Patients with high risk of developing brain mets are eligible for Part A of the study, meaning patients diagnosed with non-CNS metastasis within 24 months and with triple-negative breast cancer, HER2-positive breast cancer, non-small cell lung cancer, small cell lung cancer, and melanoma. Those patients included in Part A agreed to undergo several samplings at different time points, including neurological examination, brain MRI, NSE, and plasma samplings. Once a year for breast cancer, every 4 months for lung cancer, and every 6 months for melanoma. For patients that are developing brain mets and already included in Part A, or patients not included in the program and developing brain mets, they can enter Part B of the program. At Part B, the patient undergo mandatory CSF sampling and plasma sampling, as well as responding to quality of life assessment questionnaires. There is also an optional non-CNS tumor tissue collection, and in case of CNS tumor tissue, CNS tumor surgery, tumor tissue from the CNS is also collected at any point of the study. For Part C, the patients are followed prospectively with plasma sampling and CSF sampling if clinically indicated. At the time, 70 centers from the OncoDistinct network are participating to this initiative in France, Belgium, Luxembourg, and we are looking to extend this project to further countries. So this program allows us to constitute a clinico-pathological database and biobank for CNS metastasis, including clinico-demographic factors, imaging data, CSF and plasma samples, and CNS and extra-CNS tumor samples. And this is done with the aim to develop in each part of the program a clinical trials targeting important challenge for CNS metastasis. So in part A, primary prevention and early detection strategies, at part B, new innovative treatment strategies, and at part C, prevention and innovative treatment strategies. I now want to focus on two aspects of the program that are preliminary because it's still ongoing, but wanted to give you some more concrete results and idea today of what we're doing. As you know, few available data and guidelines on brain met screening strategies and primary preventions are available today. We know that there are discussions concerning prophylactic cranial irradiation versus MRI screening in small cell lung cancer. We know about the potential of next generation TKIs for lung cancer and primary prevention. There are some recommendations for baseline brain MRI screening in non-small cell and melanoma patients, but otherwise no clear guidelines today. So we believe that there is a need to develop predictive models for CNS metastasis to better select the population eligible for systematic screening and to help the development of prevention and early detection clinical trials. So currently there are clinical calculators that are being developed as the one we have developed in collaboration with MD Anderson in early breast cancer patients, and that allows to identify key risk factors, clinical key risk factors for brain mets. But still, we believe that combining imaging features with clinical and biological data could enable the construction of more accurate predictive model for brain mets and guiding more precise treatment and improve prognosis of the patients. So as part of the Brainstorm program, we have evaluated the role of serum neuron-specific enolase as a biomarker for CNS metastasis. Neuron-specific enolase is expressed in the human brain and measurable upon brain damage in the blood serum. It has been investigated previously as a biomarker of brain damage and intracranial response to treatment with no clear conclusion. We have hypothesized that it may contribute as part of other predictive models to the early detection of CNS metastasis. So in Brainstorm, patients undergo NSE samplings in part A and part B to assess the predictive value of NSE for the development of CNS metastasis. Very early results were presented at the MAP conference in 2023. So at that time, 34 patients were included with NSE levels available, 26 in part A and 8 in part B. And it appears that median NSE was numerically higher in part B, and that it was positive for 62.5% of patients in part B as compared to 34.6% of patients in part A. So this underlines the trend for higher serum NSE levels in patients with established CNS metastasis and supporting the need to formally investigate NSE as a non-invasive circulating biomarker of CNS metastasis in correlation with other approaches, of course. We also use, and I saw you have a similar project, retrospective data from the Institut Jules Bordet, comprising 115 patients with small cell lung cancer and 576 patients with non-small cell lung cancer. Out of them, 323 patients had brain mets, to develop a predictive AI-driven model for brain metastasis based on baseline thoracic CT and brain MRI available for those patients, and in combination with clinical variables. This radiomix-based predictive model will be validated prospectively in the Brainstorm program and correlated with plasma ctDNA and NSE findings from the Brainstorm program. A second project of the Brainstorm program is to better understand biology of CNS metastasis using CSF ctDNA as a surrogate endpoint for CNS tumor tissue DNA. Of course, you know that CNS metastases have a molecular branched evolution, and that it has been demonstrated already 10 years ago by Brastanos and all that brain metastases have actionable mutations that are not always detected in primary tumor or extra-CNS metastases, and that they are sharing actionable driver alterations that cannot always be captured by genotyping the primary or the extracranial metastatic sites. So brain biopsies are still often considered as an invasive approach, and less patients are undergoing CNS surgery. So CSF ctDNA is now investigated as a surrogate for brain met DNA. And it has been already demonstrated that CSF ctDNA better represents the genomic alteration of brain tumors than plasma. This is a publication that was from 2015, and since then all other publications came with the same conclusion. And more recently, this comprehensive analysis of CSF ctDNA in 584 patients with lung cancer and CNS metastasis was published, demonstrated that CSF ctDNA is detectable in 67.8% of patients with CNS metastasis from lung cancer, even more in presence of leptomeningeal disease, and seems to increase with CNS tumor burden. For the purpose of our analysis, we have the following endpoints. Evaluate the presence of CSF ctDNA at diagnosis of CNS metastasis. Compare CSF ctDNA mutational profile with matched tumor tissue and plasma mutational profile. Assess concordance of mutational profiles among overall population and in patients with leptomeningeal metastasis. And finally, to detect actionable mutation in CSF ctDNA and or CNS metastasis tumor DNA. For this, we are using the following method. Tumor DNA is profiled with an OncoDeep 638 gene NGS panel, and matched plasma and CSF ctDNA undergo sequencing with the OncoFollow panel, which combine up to 15 patient-informed variants from tissue with a 50-gene hotspot panel of main cancer drivers. Pathogenic and likely pathogenic driver mutations are then cataloged as shared or compartment-specific. Then we have quantified overlap between compartments. Dr. Areco presented at ESMO this year the first results for 25 patients of this analysis. For the purpose of this analysis, all patients included in Brainstorm Part B with available baseline tumor tissue as well as plasma and CSF samples were included in the analysis. Unfortunately, only 25 patients had matched CSF and plasma samplings that were available for analysis. The median age of patients included among those 25 patients was 53 years old. As you can see here, 50% of the patients had only brain metastasis, and 44% of the patients had additional leptomeningeal disease. Number of metastatic sites identified before inclusion was 2, and the time between diagnosis and brain metastasis was approximately 3.2 years. Results are the following. ctDNA was detectable in 84% of evaluable CSF samples overall, and 91% of CSF samples in the leptomeningeal cohort. Regarding detection of mutation across compartments, we had 91% in tissue, 47% in plasma, and 49% in CSF. Interestingly, variable allele frequency in CSF seems to be a bit higher than in plasma, both in overall population and in leptomeningeal disease. Regarding mutation concordance at the patient level in the overall population, we had 52% between tissue and plasma, 45% between tissue and CSF, and 75% between plasma and CSF. Interestingly, one TP53 mutation was detected in plasma and CSF only, and two TP53 mutations were detected in CSF only. Same kind of results were found for the leptomeningeal cohort with a patient-level concordance of 39% between tissue and plasma, 42% between tissue and CSF, and 91% between plasma and CSF, with again one TP53 mutation detected in CSF only. So our study presented at ESMO had some limitations, a small sample size, but also, unfortunately, the missing matched tumor tissue for brain mets, as very few patients had surgery at the moment of inclusion. But overall, CSF ctDNA was detectable in patients with both brain and leptomeningeal metastasis and showed stronger concordance with plasma ctDNA and only partial overlap with tumor tissue. CSF ctDNA also uncovered CNS unspecific mutations that were not captured in solid tumors or plasma, and underscoring its complementary role for molecular profiling and its potential to guide targeted strategies in both CNS metastasis diagnosis and treatments. So what are our future directions within the Brainstorm program? We plan to further expand the data and sample collection and pursue the ongoing research I've just presented, but also explore opportunities for Part C with epidemiological study, prevention, and treatment studies, harmonize with national and international efforts, and we're very willing to create collaboration opportunities, and also implement clinical trial within the program. And I wanted to finish my talk with two concrete examples of studies that we plan to implement within the platform. Absinthe is a clinical stage molecular imaging company. They are developing in vivo diagnostics empowered by single domain antibodies, also called nanobodies, that can be used for easy radio labeling. And they have developed a tracer called ABS011 and HER2 tracers that can be used for HER2 PET purposes. In a phase 2 trial, they have demonstrated that their HER2 PET is able to have uptake in breast cancer brain metastasis, both HER2 positive and HER2 negative. They also show that it could be used to detect HER2 low and positive brain mets earlier and to evaluate the potential progression after radiotherapy. So in Brainstorm, we aim to implement an exploratory sub-study including approximately 20 patients from Part C of the Brainstorm trial. The main objective would be to assess the discordance between intra- and extracerebral HER2 PET imaging in a population of patients with breast cancer brain metastasis from any subtype and about to initiate treatment with TDXD. The secondary objective would be to evaluate the correlation between HER2 PET findings in CSF and treatment response. Patients with breast cancer brain metastasis with or without leptomeningeal involvement would be eligible. They could have prior treatment to brain mets, but needs to be candidate to 40DXD. They would have a HER2 PET baseline, and this HER2 PET would be then redo after cycle 3 of TDXD. Finally, one of the ultimate goals of the Brainstorm program would be to be able to implement an umbrella trial for CNS metastasis based on the molecular abnormalities that we would have identified in the CSF and or the tumor tissue whenever available. With that, I would like to thank everyone who participated to the Brainstorm program, including patients and families, Françoise Roté, Professor Awada, and the BCTL lab, as well as the CTCE team and OncoDNA. Thank you.
Is there any question in the audience? So I'm a little bit now puzzled with the CSF to plasma similarities. It's quite high in your studies, no?
Yeah.
Even in the lepto and in the parenchymal metastasis.
Yeah. It's a bit less when there is no leptomeningeal involvement. I think it makes sense. One of the explanation might also be that for the tumor tissue that we have used, it was archival tumor tissue most of the time from the moment of the diagnosis for mostly breast cancer patients. So it could be like 10 years before. So I believe this might be the reason why there is a huge difference between what we see plasma CSF that are taken at the same time and also the tumor tissue. And another thing is that this information we have to get. We have to see what was the extent of the extraCNS disease at the moment of the sampling. So this is something we are looking at.
Yeah, Aurelio. Thank you.
Thank you. My question is in the same direction, Manuel. So when you are talking about tissue, you are talking about the primary tumor, or you are talking the brain metastasis tissue? Because
yeah, both of them. Because for the purpose of the protocol, as we are requesting archival tumor tissue with no mandatory biopsy for funding reasons, because it's an academic trial, we had to accept both. So here for the 25 patients, most of the patients, it's primary tissue and 1/3 metastasis. So it's a mixture of both.
But if you compare the tissue from the brain metastasis?
No, for the purpose of this analysis, we had no one undergo surgery except for one patient. So it wasn't sufficient to report here.
Thank you.
Wonderful presentation, a wonderful effort. And for the umbrella trial that you are planning, are you planning to select patients based on the CSF results or in the tumor results? Because when you are using a tumor-informed method, it takes a while to have all the information and then check the CSF. But that could be a bit a challenge, because you have the information from the CSF and the tumor and the plasma.
You mean the timing from, yes, so the proposition would be either to use, if we have already information from outside CNS, maybe not exclude those patients if there is a need to be treated rapidly. Otherwise, if patients undergo this CSF sampling and tumor tissue analysis at the time of entering Part B, the umbrella trial could be implemented also in Part C, so at any time that they would need it.
Can I ask? I have. So what about low-risk primary tumors that end up developing brain mets? No, because
there was a few, in fact. It was when you see, so I don't have the number here, but I was quite struck by the fact that almost half of the patients that are entering Part B had luminal breast cancer, luminal A, luminal B breast cancer, so it was a bit surprising, but it also made me think that it was a good idea to add this specific cohort to enable the possibility to enter directly in Part B. Therefore, maybe we're going to understand a bit better why it happened.
And just maybe to finish, so what is your vision about the real possibility to prevent brain metastasis? I mean, the example of lorlatinib is really amazing.
Yeah.
This seems to be possible. What do you think? There are papers like the paper from Gelares in terms of this methylome from stage 1 lung cancer in blood that they can predict who is going to develop brain mets. How is this going to evolve?
Yeah, I hope positively. So we're looking forward to that. It's clearly a complicated question because I believe this also might be dependent of the tumor type. So both the prevention strategy, but also the way to detect, to predict the brain metastasis. So maybe something that could be easier at the first step and more achievable would be early detection, to allow also to treat more earlier and with less invasive procedures. But still, it's work ongoing.
Thank you very much, Nuria. Thank you for coming.
Ernest is an oncologist and also a researcher, a medical researcher, that has been studying for a number of years the lung cancer and mesothelioma tumors. He is working in the ICO, in the Instituto Nacional de Oncología, in Barcelona, in L'Hospitalet. And he is the leader of the section, he has been the leader of the section of thoracic tumors, and now is currently coordinating the 3 hospitals for these thoracic tumors. And also, he has been appointed as Director of Research and Innovation of the ICO at the beginning of this year. So it's a pleasure to be here. Thank you so much for this opportunity to listen to you. And
thank you so much. I would like to thank the organizing committee for inviting me. It's a real pleasure to be here today, and I was really impressed by all the efforts that you did in the RENACER. I will be talking, this talk is going to be a bit more focused on lung cancer as a disease, and the title was the challenges, but I will change a bit. I will cover some of the challenges, but I will give you some results that we have generated during the last years too, and I hope that I will be happy to take some questions at the end. These are my conflicts, and I would like to start expressing our concern because brain metastasis is a common complication in patients with lung cancer because it's really, the incidence is higher than in other tumor types, and as you know, this is associated with dismal prognosis, and once the patient develops brain metastasis, there is a huge impact in the quality of life. And the management of these patients is becoming more complex during the last years because we have more opportunities and more therapeutic approaches to those patients. And managing those patients is really complex because there are many factors that we need to take into account. I think the audience is mixed. There are some clinicians, but there are a lot of people from the lab working in this, focusing on brain metastasis. And when a clinician has one patient in front of us, I think it's really important to understand all this complexity. Some of the characteristics are related with how the tumor is presenting. Others could be, which is the histology and the PD-L1 expression, and the genomics information is becoming more and more important. And also, we took into account which are the treatments that patient received, if that patient could develop or not resistance to immunotherapy or targeted therapies. And it's really important also to take into account which are the patient preferences and which is the functional status of that patient and the comorbidities. I think we need to place the patient in the center, and it's important. I'd like to say that genomic medicine is beyond, personalized medicine goes beyond genomics, and it's really important to personalize our treatments, taking into account all these factors. And I like to say that when we have a patient with brain metastasis, we don't have one disease, we have 2 diseases. And it's really important to maintain the balance because if we start treating patients with a huge tumor burden out of the brain with radiation, for example, there is a risk that that patient could deteriorate and we can lose that patient. On the other hand, there are patients that we could, and then we can decide to start systemic therapy, but if we don't control well the CNS disease, that patient can deteriorate too. Then we have 2 major fronts, and we need to manage these patients properly. And it's really important to discuss those patients that should be managed always by a multidisciplinary team, in a multidisciplinary board, or in a committee that we have the expertise from radiation oncology, neurosurgeons, medical oncologists, because this is the best way to go to individualize the decisions. And one challenge we have when we have to decide what to do in the clinic is that patients with brain metastasis, but not in lung cancer, in all the cancers, they have been generally underrepresented in many trials. And there are several reasons for that, because these patients generally they have poor performance status, they have limited life expectancy. Many of them are receiving corticosteroids. That has been a limitation for immunotherapy for many years. We know that the blood-brain barrier is a major challenge for some drugs, and some patients also are difficult to assess when we have, because we may have some dissociated responses or pseudoprogression, and that was the reason why pharma companies many times exclude excluded patients, or they are really stringent when we are selecting patients for investigating new drugs. And that's a challenge because we have then to decide in the clinic and we have limitations. But the guidelines are quite aligned in terms of management of lung cancer patients with lung cancer with brain metastases. I want to use patient-centered language. And according to the American and the European guidelines, you can see that they can be simplified to when we have a patient with symptomatic CNS lesions, we should consider local therapy, surgery, SRS, stereotactic radiosurgery, or whole brain radiotherapy. And when patients are asymptomatic or the symptoms can be controlled with medical treatment like corticosteroids, then we can consider to defer the local therapy. But that should be always decided by a dedicated MDT, multidisciplinary team. And in patients that they have genomic alterations that they could be treated with drugs that are able to penetrate into the CNS, then we can consider to start those drugs and defer the radiation therapy. There was a lot of controversial discussions if we could do that also in patients treated with immunotherapy, but during the last years we have learned that patients with brain metastases, even when they didn't receive local therapy, they can be also treated safely with immunotherapy, and we can achieve a significant activity in the brain. I would like also to bring, I know that there are some radiation oncologists in the room, and we can discuss that later, but I think they are also really aligned with that view that whole brain radiotherapy is a very old treatment that we still use in some patients, but it has been demonstrated in several studies that this approach is very toxic for patients and the efficacy is very modest, and there is a huge impact on the quality of life, and I think we need to incorporate more modern techniques like stereotactic radiotherapy. That is probably the way to go, but it's true that there is a problem of access to this more sophisticated radiation and at least in our center, for example, we are treating patients that they have up to 7 lesions, but there are patients that they could present in the clinic with more than 10 lesions, and then that could be an issue. Particularly in a country like us, that we have a universal public health system, I think it's important to have an equity in the access to these more modern radiotherapy techniques. And as you know, whole brain radiotherapy is associated with leukoencephalopathy, and it has a major impact in the neurocognitive function in the patients. And despite there are some mitigation strategies to minimize this neurocognitive impairment, like the use of memantine or hippocampal avoidance, there is an issue in those patients that after significant follow-up, they could develop neurocognitive impairment. I think the major difference has been also that in the past, patients with lung cancer, they live for a few months when they have brain metastases, but now with the new drugs that we have, they could live many years. Then we are becoming more and more concerned about the long-term toxicities. And also in lung cancer, it's really important to establish a genomic testing. The genomic testing is really important, and the molecular diagnosis is a major issue still in the world. And I think it's important to establish which are the main alterations for patients with non-small cell lung cancer, because there are, at this moment, there are at least 10 different molecular alterations that can be treated with specific targeted therapies. And some of them, they can achieve significant efficacy in the brain. Sorry that the slide is a bit fuzzy, but you can see that there are several drugs here in the I think that's difficult to manage, but there are distinct drugs that they have very high penetrance in the brain, that they have been already approved for different alterations like EGFR, ALK, ROS1, or RET. And you can see in blue, which is the CNS activity that is even higher than in the body for some of these drugs. For some drugs like lorlatinib, that was already mentioned by Nuria before, the complete response rate is really high. But we have another drugs that are on the right that despite they achieve efficacy in the brain, they are not so active. And I think it's important that we need to distinguish. We cannot put all the alterations in the same bucket. For some alterations, we can initiate TKI therapy, targeted therapy, to control the CNS disease, and then we can defer radiation therapy, but there are another alterations that we should be more cautious, and we need to discuss those patients in the multidisciplinary tumor board to decide which is the best approach for those patients. Because if we assimilate all the patients in the same group, we may have some patients failing quite early to these targeted therapies, and I think it's important to take that into account. During the last years, I don't want to focus a lot on that, but several strategies have been explored to improve the efficacy in the brain in patients that they have lung cancer and targeted therapies, for example, increasing the dose of the TKI or incorporating stereotactic radiotherapy in addition to the targeted therapy. And now, for example, for EGFR mutant non-small cell lung cancer, we have a couple of regimens that they combine osimertinib plus chemotherapy or lazertinib with a bispecific targeting EGFR and MET called amivantamab, that they demonstrated a significant improvement in survival in patients that they have brain metastases. That means that the patients, when they are fit, we can intensify the therapy to improve the control of the disease in the brain. But I would like to talk today more about which are the challenges and the opportunities in patients with brain metastases when they are not candidates for these targeted therapies that they have been. We have seen amazing efficacy, but there is a more difficult scenario that when these patients are not eligible for targeted therapies, most of them they receive immunotherapy alone or immunotherapy combined with chemotherapy. During years, we know that the brain has been considered as an area that is or a sanctuary where the immune system cannot be activated, but we learned first from our melanoma colleagues that this is not truly like this, and also in patients with brain metastases coming from the lung cancer and breast cancer and other tumor types, we have seen that the immunotherapy can be effective. I don't want to go to all these studies, but this is just to show that we have two groups of studies. The data that is demonstrating that immunotherapy is effective in the brain is coming from two major groups of studies. One at the top you can see clinical trials sponsored mainly by pharma companies, that they were quite selective in terms of allowing patients with brain metastases to go into the study, because most of these studies, they request that the brain lesion should be previously treated with local therapy, surgery or radiation therapy. And we have a post-hoc analysis showing that, I will show you later, that from those studies that immunotherapy is effective and is able to control the CNS disease and is improving overall survival in those patients. On the other hand, we have academic studies that are dedicated brain metastasis studies that mainly, most of them are coming from comparative groups, but there are others also that have been sponsored by pharma that became a bit more open, and they allow patients to be treated within those studies even if they didn't receive prior local therapy. And focusing first in the first group of studies, those that they were sponsored by the pharma companies, and they were not designed to respond this question of immunotherapy, to know if immunotherapy is working well in patients with brain metastases. Despite these limitations, they enroll patients with previously treated brain metastases, and we have seen in these different studies that immunotherapy, when is combined with chemotherapy or is given alone, is better than the old approach that was given, that consisted on giving only chemotherapy. And they demonstrated in all these different studies, it was demonstrated that there is an improvement in the overall survival. We have also data that we will dissect a bit more, showing that also in patients that they didn't receive prior local therapy, immunotherapy is effective in those patients. And the first study and the first proof of concept is coming from the Yale University. I was on a study in patients with non-small cell lung cancer that they have brain metastases, and pembrolizumab, that is an anti-PD-1, was the treatment that the patients received, and the intracranial response rate was 30%. And there were some complete responses, and the intracranial PFS was a bit disappointing, only 2 months, and the median overall survival was 9.9 months. I think the study demonstrated that there is efficacy, but the duration of the control of the disease is quite short. We in our group we had a lot of interest in patients with brain metastases, and we were able to design an academic study called Ateso Brain in collaboration with the Spanish Lung Cancer Group, and we were concerned about patients with non-small cell lung cancer with brain metastases, and we were quite concerned because a few years ago when we designed this study, whole brain radiotherapy was the main treatment for many patients in our country, and we have that feeling that many of these patients, they could deteriorate during the whole brain radiotherapy, and some of them, they were not able to receive systemic therapy with chemoimmunotherapy. The study was quite open. We allowed patients with non-squamous, non-small cell lung cancer, they were EGFR and ALK negative, any PD-L1, they should have measurable brain lesions, and we allow patients to receive low-dose dexamethasone or medium-dose dexamethasone that was up to 4 milligrams per day. That many of the studies that I commented that were sponsored by the pharma, they were really selective, and these patients were excluded from those studies. Patients received a combination of chemotherapy plus atezolizumab and anti-PD-L1, and the primary endpoints were to see if this combination was safe for patients and which was the efficacy, particularly in the brain, using RANO brain metastases. We found that the intracranial response rate using this combination was 40%, and we had 4 patients with complete responses. I would like to remind that those patients didn't receive radiation therapy, and 15% of the patients had a dissociative response. That means that they responded in the brain, but there was a progression in the body or the other way around. And the median duration of the response in the CNS was 14 months. That is not bad, but it's true that we would like to perform better. And the median intracranial progression-free survival, that means the time that the disease is failing to our treatment in the brain, was 6.9 months compared with 8.9 months in the rest of the body. That means that it's a bit behind, but it's quite similar with the efficacy in the other compartment. And I would like to also recognize that 60% of patients receive brain radiation when they progress, mainly whole brain radiotherapy, because in many centers they don't have access to stereotactic radiotherapy. And the median time from treatment initiation to brain radiotherapy was about 11 months. And that was one result that was for us something that make us think, or we are thinking on that result, because we consider that now probably what we have to do is integrate better radiation therapy with systemic therapy. In that study, we have also a lot of interest in understanding which is the impact of the treatment in the neurocognitive function of the patients. And we conducted, in collaboration with neuro-oncologists, a prospective study within the clinical trial to analyze which is the impact of the treatment. And we have seen that patients that they responded well to chemoimmunotherapy, they have an improvement in some of the indicators, like, for example, verbal fluency measured by one validated test. And also in patients that they didn't respond, they have a verbal memory decline. And these are studies that are difficult to perform, but I think are important to demonstrate that the use of systemic therapy can control the disease, and it seems that it doesn't have a major impact in the cognitive function. We are also conducting some studies using the brain MRIs collected within the study, trying to identify radiological features that are associated with benefit or lack of benefit in within the study. And we found that when we compare patients that responded well to the treatment compared with those that were resistant, we found that patients that they responded, they have lower tumor burden in the brain. And also we identified a marker that is when we see the flare compared to the flare means the inflammation that you can see in one of the sequences of the brain MRI. The general is easy to capture that information compared to the other sequence that is the T1 gadolinium, that it helps you to identify which is the volume. We found that this ratio was helpful to identify patients that they have worse response to the treatment and worse overall survival. Obviously, this was conducted in a very small cohort, 40 patients, and now we want to validate those markers in larger series. We also presented just 1 month ago in the World Congress of Lung Cancer another study that we conducted in the Spanish Lung Cancer Group that is called VP Brain. In this case, it was a similar population of patients with advanced non-small cell lung cancer. In this study, we also allow patients with squamous cell carcinoma, and all patients, they have untreated brain metastases that were not amenable for stereotactic radiotherapy. And we have 2 cohorts. We have one cohort, cohort A, with asymptomatic brain metastases that were not receiving steroids, and cohort B with oligosymptomatic brain metastases that patients were controlled with low dose of steroids. In this case, patients were receiving a short course of chemotherapy, only 2 cycles, and we used 2 immunotherapy drugs, nivolumab, an anti-PD-1, and ipilimumab, that is an anti-CTLA-4. And in this case, the primary endpoint was to demonstrate the intracranial control of the disease during the first 6 months, and there were a number of other secondary endpoints. We found that the intracranial response using this combination was 41%. That was really was equal than the overall response rate that we observed in the rest of the body, because we use different methods to measure the response within and out the brain compartment. And we observed also that most of the responses were concordant in both compartments. But again, we have 4 patients that they have, 5 patients in total that they have dissociated responses in the brain and in the rest of the body. This is the main result of the study. The primary endpoint was to demonstrate the intracranial disease control rate at 6 months. We were expecting at least 50%, and the study was considered negative because we fall a bit below, and we have 41% and 46% of patients free of progression in cohort A and cohort B. Probably we were too optimistic when we were expecting at least 50%. But this is a population that some of the patients, they have high burden of disease in the brain and in the rest of the body, and we know that lung cancer is a difficult disease to treat. In terms of the intracranial PFS, the median intracranial PFS in this study was 5 months, a bit shorter than in the first study that we conducted, and the systemic PFS was 6.7 months. In terms of overall survival, the median overall survival was about 11 months, but the 2-year overall survival rate was 29%. You can see that there is a plateau and a group of patients that are long-term survivors. And we were quite surprised by this result, because I would like to remember that patients in cohort A were not receiving steroids, while patients in cohort B, they were receiving dexamethasone, and there is no differences in terms of overall survival according to these when we compare these 2 groups, and this is something that it was a bit controversial for us. The study was not designed to answer this question, but we were expecting worse survival for patients within cohort B. That means that probably it's true that patients that are receiving steroids, they are more symptomatic, they have more burden of disease in the brain, but probably it's more complex than that, and there are some patients receiving steroids that they could respond really well to immunotherapy combined with chemotherapy. In terms of safety, we didn't observe, we didn't have any toxic death, and we had some neurological adverse events. Some of them, they were grade 3 and 4, but they were only 5 patients, they have some of these toxicities, and the toxicity was expected from this combination. There are a number of other trials that have been investigating the combination of chemo plus immunotherapy. Many of them are conducted in China, and they have shown similar results, a bit better, but that always happens in many studies coming from Asia, that the results are a bit higher in terms of efficacy outcomes. Probably there are differences in terms of when we compare these studies that are all small, they may have some differences in terms of patient characteristics, but there is a big wave of studies, academic studies coming from Asia that validate those results. But I think the next question is how to combine chemoimmunotherapy or immunotherapy alone with stereotactic radiotherapy, because you have seen that the chemoimmunotherapy is active, is effective, but the problem is that along the way, the patients will progress, and we don't want to do whole brain radiotherapy because these patients progress. I think it's really important to incorporate SRS within this systemic treatment. And there are a couple of studies in Europe conducted by two cooperative groups, the ETOP and EORTC, that are trying to answer these questions, and they compare one arm that the patients receive chemoimmunotherapy alone, and other group of patients, they will receive early stereotactic radiotherapy to try to see if this approach can improve intracranial progression-free survival. And there is another concern when we incorporate stereotactic radiotherapy. One of the problems is that patients survive, and it's true that it's less toxic than whole brain radiotherapy, but some patients will develop radionecrosis. That is a complication that could be difficult to manage and can cause a lot of problems to patients. And we are still learning how to incorporate SRS with chemoimmunotherapy to reduce the incidence of this problem that we can see in patients that are long-term survivors. And I would like also to mention that there's more innovation coming beyond chemoimmunotherapy, targeted therapy, and radiation therapy. There are some physical therapies that could be also relevant. For example, there are some studies testing ultrasounds to open the blood-brain barrier, and that could be interesting when we want to deliver large molecules. And there is also the treatment fields approach that is using electromagnetic fields to try to disrupt the mitosis of the tumor cells, and it has been demonstrated in glioblastoma that can improve overall survival in patients. There are also data in brain metastases from lung cancer, but it's true that the study design of those studies is a bit controversial, and I think we need more data to demonstrate the efficacy of this approach or approaches, but I think in the next years we will have more data coming. And this is my last slide, some take-home messages that I would like to remind. I think it's important to avoid whole brain radiotherapy as much as possible because this is a really toxic treatment for patients. I think multidisciplinary approach is crucial, and we need always to discuss those patients within a tumor board. It is also important to reduce the dose of steroids to the minimum that we need. Also, I will advocate for enrolling patients with brain metastasis in clinical trials, and also it's important to do some lobby with pharma to have trials that are more open-minded and are able to enroll more patients with these characteristics because now they are still really restrictive. We know that immunotherapy is working in the CNS, despite all the skepticism that we have many years ago. One of the major questions now is how to integrate immunotherapy with stereotactic radiotherapy. And also we need to improve our basic knowledge in the CNS metastases to design better trials. That is one of the aims that you have in RENACER. And I would like to finally thank all the team that I have the privilege to work with. Thank you so much.
So is there any question for Ernest in the audience?
So great overview. It's not a question, it's just a comment, and it's that I fully agree with you with your presentation, so we can be friends.
That's great. No, because when there are some radiation oncologists in the room and you are trying to, I don't say kill, but to make some criticisms about whole brain. No, but the problem, I think, is the major problem in Spain is the access to SRS. That we know that in the private hospital there is major, they have everything organized, but in the public hospitals, the number of patients that are getting access to those therapies, at the end you need a criteria, because if not, the number will be, you cannot assume that. And we started treating three lesions many years ago. Now we moved to 5, 7, but it depends many days on which is the burden that we have. But we have seen in the US centers treating up to 15 lesions. I think we need to become more open-minded, but probably we need also to press, to make lobby, to have bigger teams in the hospitals to be able to offer the best treatment to a larger population of patients.
So, Ernest, thank you very much. Very interesting. I mean, you know the numbers. So how many brain metastases do not have actionable vulnerabilities? You mentioned that there are about 10 that have associated drugs that you can test, but what are the numbers? I mean, what is the percentage of patients that do not qualify
The number of patients that are able to receive one of these targeted therapies is about 40% nowadays. 40%. Still a bit more than half, and also in Spain we have some restrictions to some of these drugs that they appear in the recommendations by the European guidelines. We still don't have access to all these drugs in the front line. But if we put all them together, will be 40% of patients. Still, 50 to 60%, they receive mainly chemoimmunotherapy or immunotherapy in the front line.
And in terms of the efforts, because of course, I imagine that for those that do not have any of those genomic alterations, then the main hope is the immunotherapy, I guess. But, from your experience, how can we develop new therapies? What is the strategy? I mean, it's a very complex population of patients, getting more complex because you have different lines within a standard of care. How we can put more drugs in clinical trials for brain metastasis? I know it's a very...
but I love that question because we, as Nuria also said, it's really challenging because pharma companies, they are really restrictive because when they have registrational studies, they want to approve the drug. Then they don't want too many patients with poor prognosis within the study. And obviously it's a problem. If the patient deteriorates, you don't have the data about that patient. But we have seen a bit of change during the last years. I think these trials helped to be a bit more open, and they are now allowing patients with untreated, so not everybody, not all the companies, but now they allow patients with untreated brain lesions where they are asymptomatic. That in the past was like a dogma, and that changed during the last years. But they are always a minority because some of these patients are symptomatic, they need some local therapy, and some of them, they need time to recover, and there are patients that there is a big attrition. One of the problems we have also in patients with brain metastasis is that once you deliver local therapy, they need to recover, you need to reduce the dose of steroids, and some of these patients that are not able to reach the trials. This is why we wanted to design these studies to start the systemic therapy first and delay a bit the local therapy. And the other way to go, in addition to push pharma companies to be more open-minded and to allow more patients with brain metastasis within the studies, is to design academic studies or dedicated brain studies. The problem is when you request money for those studies, it's really difficult to conduct a phase 3 study. Generally, you are able to design a small study that is a phase 2 that is able to generate some efficacy and safety data, and you can do then some correlative studies with imaging or another type of biomarkers, but it's true that the number of patients is small, and then we have the issue that then you don't have a lot of statistical power to make big questions. No? And then we have these contradictory scenarios that are challenging. On the other hand, I think it would be nice that the pharma companies could pull more data together, no, to understand, because now there is some post-hoc analysis, but we never have seen a meta-analysis of this data altogether, but this is always difficult, no? When and on the other hand, it's a post-hoc analysis that was not planned, and then there is many confounding factors that they could also be complicated when we want to analyze those results. But I think we need to push in both directions, to pharma to have more open studies, and also to pharma and cooperate more, to have these cooperative initiatives and investigator-initiated studies that are completely dedicated to brain, because also these dedicated studies allow you to use random criteria, to have radiomics, focusing on those patients, and there are opportunities I think they create opportunities for learning more, as you said, which is the best way to go and to continue.
Thank you.
Just a curiosity, how is the neurocognitive testing, how is this done in the clinical trial? I'm not familiar at all with clinical trials. So.
Yeah, it depends. In our hospital, we have a strong neuro-oncology clinic, and are the neurologists who are doing that, because there is one person that is dedicated to this kind of projects. But when we run those studies in Spain, we have some issues, and we need. There were some hospitals that have neuropsychologists. We have been also doing some training from our team. We did several trainings to the different hospitals, particularly with those that they have limited experience. And we supported also them when they were doing all the, for example, there are some questionnaires that are difficult to interpret, and then we were providing some support. When they have some issues, we organized a meeting, and there was a lot of dedication from our neuro-oncologist. But on the other hand, we have also similar challenges like you, that some patients, they comply with the procedures, particularly at baseline, the first assessment, but we don't have many assessments in later time points. That happens always also with quality of life assessments, but here is even a bit more difficult because they are difficult to perform and time consuming, and then many times we have so many patients coming to the clinic that this is quite challenging.
Thank you. Any other question?
Hi, Ernest. Very nice talk. I've learned a lot. I wanted to ask you, how do you measure intracranial progression? Like how do you define that a patient has progressed, because it's a problem that we are having in our cohort here.
In the mice? No, in your cohort, in your clinical cohort, okay.
Yeah, in RENACER.
Yeah. It's true that in the two academic studies that I commented, we use RANO criteria, and then it's defined by the size of the lesions, and it takes into account also some clinical features, and like if the patient is also deteriorating, it's something that you need to take into account. But mainly it's the size of the lesions that we have been using. But it's true that it's really challenging. When we have been reviewing the brain MRIs in that radiology, in the radiomics project, we have seen some discordance, and I think it's difficult to assess the response, particularly when you want to use RECIST or RANO, because in the clinic, when we are not in a clinical study, in general, medical oncologists and radiation oncologists, we don't have time to do RECIST and RANO, and that's an issue then, because you want to collect that. And then probably it's quite, out of a clinical study, the decision of progression is quite qualitative, and it's based on the opinion of the neuroradiologist. Then you are not capturing the dimensions, because that will be, you will need a lot of dedication. You need to decide several lesions and measure them along the way and see if they grow more than 20%. We consider that, or the appearance of new lesions. But probably what is easy to capture is when there are new lesions. But when the criteria of more than 20% that is used by RANO and RECIST is something that you will probably don't have, because the reality is that we don't use these scales and these criteria in the clinical practice. But I think we should push more our radiologists to do that. And also probably in a few years with the evolution of artificial intelligence, probably will be a bit easier for the radiologist to get this information in a faster way, because at this moment the problem is a problem of hands and time, that the radiologists are under a lot of work. But a good point.
Yeah.
So when you're talking to driver mutation, you're talking about the primary. But if you compare when you have tissue from the metastasis, if you compare both, sometimes they lose the driver mutation or the PDL1. So what is there is gain of mutations or loss of mutation? What is your experience with brain metastasis?
See, in the studies I presented, these were patients with advanced disease, and mainly we have biopsies coming from the lung or from one metastatic lesion out of the brain. We didn't have tissue from the brain, and we don't have CSF. We have blood from those patients, but it's true that the studies that were published show or reported what you say, that there is a heterogeneity between the primary lesion and the brain lesion. We have been working in collaboration with Kurt Schalper, that is a pathologist from Yale. We have one of the medical oncologists from the team has been working with him during 1 year, and she has been analyzing a couple of TMAs from our institution and from Yale, and we have seen important differences when we check some biomarkers, and this still, we will publish this during the next year, but we have seen, for example, some differences in terms of some primary resistance features to immunotherapy. For example, the antigen-presenting machinery is generally more affected in the brain than in the rest of the body, and that explains why these patients, they don't respond well to immunotherapy in some cases. But it's true that this is one challenge that we have. Probably with the CSF analysis, we will be able, because only a minority of patients with brain metastasis are resected, mainly when they have only two scenarios. I didn't talk about that. When they have one lesion and you want to be sure that that's coming from the primary tumor, or you don't have any tumor visible in the body and you have a brain metastasis, that is really unusual, but that happens. Or when you have a lesion that is poorly located, that is causing big problems to that patient, and you know that with a surgery you can solve that complication, and it depends on the locations. Radiation could be very challenging, and then we have these particular scenarios. Then at the end we have tumor in our biobanks, but that coming from patients that have been resected, but it's a minority, and there is a strong bias, because this does not represent the majority of patients that we are seeing in the clinic. On the other hand, I love what Nuria showed about the CSF, but CSF is still very invasive for patients, because we need to do a lumbar puncture, and I think it will be nice to have another approaches, but I think we know that the blood is falling close, but is not measuring the same type of alterations, and is probably the best way to understand this heterogeneity at this moment. And also probably imaging will evolve, and we will have. There are some studies that are trying to understand in the lung. If the radiomics can help to identify those patients with driver alterations, but probably that will be really difficult in the brain, but who knows? I think with the radiomics evolution, maybe we can be able to capture more information from other lesions that are more difficult to access.
So any other question? So thank you very much.
Thank you so much. Thank you.
Bueno, hemos terminado las charlas de los invited speakers. Ahora solemos hacer 2 charlas de speakers invitados fuera de RENACER, que esperemos que pronto se unan a RENACER. Y ahora tenemos charlas de miembros de RENACER a los que hemos invitado. Vamos a empezar la primera charla con la doctora Pilar Nicolás. Pilar Nicolás es doctora investigadora del Departamento de Derecho Público y coordinadora de la red de Cátedra de Derecho y Genoma Humano de la Universidad del País Vasco. Como veis, es una parte que a lo mejor no esperáis ver en una asamblea de metástasis cerebral, pero la realidad es que este es un pilar absolutamente fundamental para hacer lo que estamos haciendo y para mirar al futuro con tranquilidad y con visión a nivel del espacio europeo, ¿no? Pilar es coordinadora o miembro del equipo de diversos proyectos de investigación como Huracán, Eucanimage, Genome of Europe, con implicaciones en múltiples aspectos que son cruciales, como os digo, como la protección jurídica de la salud y la información genética, el consejo genético, las implicaciones legales del uso de muestras biológicas humanas y biobancos en investigación científica, Big Data, inteligencia artificial, ciberseguridad, emergencias sanitarias e intercambio internacional de datos. Como os digo, todo esto casi es algo que usamos y que necesitamos conocer a diario en RENACER. Ha estado implicada en la redacción de legislación española relacionada con la investigación biomédica y ha sido o es parte del Comité de Ética de la UPV y del Instituto de San Carlos III. Trabaja en la Junta Directiva de la Asociación Nacional de Comités de Ética de Investigación, Farmaindustria, Impact, Global Alliance for Genomics and Health, el BBMRI y, por supuesto, lo más relevante es que es miembro de la dirección de RENACER. Sin más dilación, agradezco a Pilar Nicolás el esfuerzo que ha hecho en venir aquí y muchísimas gracias.
Bueno, muchas gracias por la presentación, por la invitación y gracias también a todo el equipo de RENACER por su acogida y por haber organizado esta asamblea. Yo voy a ser muy breve y luego, como hay un espacio para preguntas, pues podremos profundizar. Me he preparado la presentación en español, pero we can go deeper in English afterwards if you have any doubt about, for example, international regulation or something like that. Bueno, voy a hacer un planteamiento muy general de el panorama normativo al que se enfrenta RENACER, ¿no? Y en general cualquier proyecto de investigación con un mínimo de complejidad ahora. RENACER se ha apoyado mucho en un biobanco, creo que es una decisión muy acertada y vamos a ver por qué. Porque en España los biobancos ofrecen un régimen flexible para el tratamiento de las muestras y de los datos, porque a la vez ofrecen también muchas garantías en la gestión. Y voy a terminar con una mirada hacia la posible expansión de las redes, de los consorcios, hacia otros países. Si decimos que son europeos es todo mucho más fácil. Si hablamos de terceros países, la cosa se complica mucho. Y no podemos dejar de hacer un pequeño recuerdo al espacio europeo de datos de salud. El reglamento, es un reglamento europeo que regula este espacio, es de marzo de este año, pero su implementación va a ser progresiva a lo largo de muchos años, más allá del año 30, porque es mucha la complicación que representa la implantación de este reglamento, mucho trabajo por hacer, se va a necesitar mucho tiempo para que se haga una realidad. Sin querer asustar, y por eso no he puesto nombres de leyes ni nada, sí que me parece necesario hacer así como una imagen que parece que intenta simplificar, explicar a qué marco normativo nos estamos enfrentando cuando hablamos de un proyecto, de un consorcio como RENACER, o de otro que sea parecido, ¿no? La normativa en España y en otros países europeos también, que regula todas las actividades que se tienen que llevar a cabo en un proyecto, esa normativa está muy segmentada, es muy sectorial, aunque se hable de un sector de actividad como el de la investigación biomédica. No hay una ley, un reglamento, no es así. Si se utilizan muestras para investigación, hay que ir a una normativa específica que regula este procedimiento, que además ha sido desarrollada por un Real Decreto. Si uno se apoya en un biobanco, lo mismo, tiene que ir a la normativa específica, bueno, además de a la normativa interna del biobanco, etc. Si se producen investigaciones invasivas en sujetos o en personas, tenemos también una regulación específica y una necesidad de evaluar determinados aspectos. Si se van a realizar en el contexto del proyecto ensayos clínicos o se van a producir, producir productos sanitarios, por ejemplo, un software de diagnóstico, o si se va a realizar un estudio observacional, esto está en otra normativa, con otros procedimientos. Y si se pretende una interacción con la práctica clínica o una conexión con medidas de salud pública, pues es que tenemos que ir a otra normativa que ya implica los derechos de los pacientes, de los ciudadanos. Y en cualquier caso, o sea, entre esto hay interacciones, pero en cualquier caso vamos a estar en el paraguas de la normativa sobre tratamiento de datos, que es una normativa global, no está pensada para un sector en particular, nos afecta en nuestra vida cuando vamos al banco, cuando utilizamos nuestro móvil en redes sociales, cuando en nuestro entorno laboral, para todos esos sectores se utiliza la misma normativa. Pasa que luego hay ciertas especificidades, pero vamos a estar siempre ahí, y además es una normativa supletoria en relación con otra más específica. Si hay algo específicamente no previsto en la normativa sobre ensayos clínicos o sobre muestras, sobre cómo gestionar los datos, entonces eso no previsto tendrá que regularse vía normativa general. Y lo mismo para la inteligencia artificial. Existe también un nuevo reglamento. Bueno, estos dos paraguas de tratamiento de datos y de inteligencia artificial están regulados por una normativa europea que además tiene la misma forma jurídica, son reglamentos. Los reglamentos tienen una aplicación directa en los estados. Se traducen a diferentes lenguas, pero el texto es el mismo y no necesita ninguna operación interna en el estado para que esté en vigor. Hay otros instrumentos jurídicos europeos que no son reglamentos y que el mecanismo de eficacia es distinto. Por ejemplo, en una directiva, que es como antes estaban regulados los datos, en una directiva es necesaria una operación en derecho interno, que se llama transposición, y a través de esa operación esa directiva se hace efectiva, pero es que esa operación supone una ley nacional que no solo traduce literalmente en términos lingüísticos, sino que reordena, reorganiza, introduce aspectos más concretos. Cuando las normas europeas son directivas, la homogeneidad en los Estados miembros se pierde. Cuando son reglamentos, la homogeneidad se intenta garantizar. Por eso estas normas son reglamentos. Otra cosa es que se haya conseguido en toda la extensión deseable. Pero vamos a acordarnos de esto porque como luego vamos a hablar de la expansión a Europa, no nos olvidemos que tenemos un marco común directamente aplicable en los mismos términos a todos los Estados miembros. Y no olvidemos también, ya que estamos hablando primero de cuestiones generales, pero muy importantes, que existen unos principios que no son principios éticos, son principios legales, que se pueden alegar ante un tribunal, por ejemplo, y que vinculan el desarrollo de toda la normativa. No puede haber una norma en contra de estos principios, ni puede haber una práctica en contra de estos principios. Parecen muy generales, pero es muy importante conocerlos, porque sirven, por ejemplo, para interpretar. Sirve para interpretar ante una duda. Muchas veces dices, si quieres dos opiniones sobre una ley, llama a dos juristas, porque uno te va a decir una y otro te va a decir la otra. Se suele decir, ¿no? Bueno, pues aparte, eso es un poco una caricatura, pero es verdad que la normativa hay que conocerla, y por eso para la interpretación de una ley es muy difícil conocer el contexto en que esa ley se publica, porque en ese contexto hay unos principios generales, hay unas conexiones con otra legislación. No nos podemos olvidar que es obligatorio que la investigación sanitaria o en salud, la investigación científica, se haga con criterios de accesibilidad, de equidad, de garantías de protección de datos, gratuidad en cuanto a que el incentivo a la participación no sea un incentivo económico, fundamentalmente se refiere a eso cuando hablamos de gratuidad. De consentimiento, es decir, que es necesario que para participar en la investigación la persona sea consciente de que está participando en ello y en qué términos y qué consecuencias tiene, y que se utilicen siempre unos datos, y yo diría, esto de la calidad va más allá de los datos, se extiende a toda la metodología en general, rigurosa y de calidad. No podemos hablar de aplicación del régimen de investigación científica a algo que no presente el rigor y la metodología propia de la ciencia. Hay muchas cuestiones en esta normativa general que se flexibilizan cuando hablamos de investigación científica para una actividad que demuestre serlo, si no, no. Pues, por ejemplo, solo para poner un ejemplo, aunque no voy a entrar a profundizar en ello, el reglamento de inteligencia artificial que contempla las herramientas de investigación de inteligencia artificial como productos, o sea, pues como un coche o como un ascensor, que cuando se pone a disposición del público tiene que cumplir unas garantías de seguridad. Es la misma filosofía. Bueno, pues excluye de su ámbito de aplicación las herramientas de inteligencia artificial que no vayan a salir del ámbito de la investigación básica, por ejemplo. Esto se puede ver como una cierta ventaja porque someterse a todos los requisitos de la regulación de inteligencia artificial no es ninguna tontería, no es fácil. Luego, hay escenarios donde los requisitos legales, las exigencias, se flexibilizan en investigación. Bueno, no en cualquier actividad de búsqueda de consentimiento, sino por un equipo acreditado, con una metodología determinada, etcétera, etcétera, y esto lo tenéis que valorar vosotros. Esto, esta evaluación no la va a hacer un jurista. O sea, el rigor de la investigación lo va a evaluar alguien que sabe de lo que está hablando. Decía que es un acierto que una red como esta, un consorcio o un programa, se fundamente en un biobanco. Un biobanco, recuerdo muy brevemente, es un establecimiento que está acreditado para eso, ha tenido que cumplir muchos requisitos. Y inspecciones, tiene que ser sostenible, tiene que tener una estructura permanente que garantiza que los derechos de las personas van a ser efectivos hoy, mañana y dentro de 5 años y de 10. Porque tiene un director científico, pero apoyado en un comité de ética externo del biobanco, externo pero conectado, y un comité científico externo del biobanco. Y entre las competencias de uno y otros comités, todos los requisitos de calidad de la ciencia y de respeto a los derechos de las personas se van a cumplir. En el régimen de utilización de las muestras en un biobanco, por supuesto, se exige información y consentimiento para la donación. Las personas donan una parte de su cuerpo al biobanco y nosotros tenemos la capacidad de disposición sobre las partes de nuestro cuerpo y por eso es necesario una expresión de nuestra voluntad libre, informada, para que ese negocio jurídico, que es la cesión, sea válido. Como todos los negocios jurídicos, tienen que estar sustentados en una voluntad válida, que se sepa que se está consintiendo, que se haga sin presión por una persona capaz, etc. Consentimiento para la donación no es lo mismo que consentimiento para tratamiento de datos. Cuando alguien dona muestras a un biobanco, esta operación está fundamentada en su consentimiento y la disponibilidad de las muestras por parte del biobanco es legal porque hay un consentimiento. Una vez que se ha consentido a la donación, el sujeto sabe que se van a extraer datos de esas muestras y que esos datos se van a utilizar para investigación, se van a ceder, etc. Y si no quiere que eso se haga, no tiene que donar la muestra. Una vez que dona la muestra, eso va a pasar. El circuito posterior de todos los datos es legal porque responde a un interés científico, al interés científico del biobanco. Lo que hace legal el tratamiento de datos en el biobanco es este interés científico. El fundamento, la base legal del tratamiento de los datos no es el consentimiento, es el interés científico de los diferentes proyectos y su solidez. Si queréis podemos profundizar en esto, pero os lo digo por si os suena lo de la base legal del tratamiento de los datos, que si es el consentimiento o es otra. No sé qué nivel de profundidad tenéis de la normativa, pero lo menciono por si queréis luego que lo comentemos más a fondo o tenéis dudas. Finalmente, el almacenamiento de las muestras se va a producir en unas condiciones de calidad técnica, no voy a entrar aquí para nada, pero se supone también que con medios humanos y de estructuras, de infraestructura que lo garantice. Y cuando esas muestras circulen, porque esto es la vocación del biobanco, circulen entre investigadores, este circuito se va a dar también en unas condiciones muy rigurosas de compromisos por las partes que transfieren y que reciben las muestras. Y todo esto una vez que se pone en marcha el biobanco, debería ser bastante sencillo y bastante ágil y bastante flexible, cosa que no ocurre en otros regímenes de utilización de muestras y de datos para investigación, donde es mucho más restrictivo su uso, porque está mucho más condicionado por el consentimiento específico que se prestó a ese proyecto por esos años para ese investigador. Esto no es así en este caso. Los biobancos son una institución que existe en muchos países, pero cuando hablamos de biobanco en término legal, es una institución española. Un biobanco en España es un biobanco acreditado por las autoridades españolas. Eso no quiere decir que la investigación también en el seno de un biobanco no se pueda expandir a otros países europeos. O conectarse con otros biobancos de otros países que tendrán su régimen jurídico correspondiente o a lo mejor no lo tendrán en ese país. Pero los biobancos españoles pueden integrar muestras procedentes de otros países. Dice la Ley de Investigación Biomédica que se pueden integrar muestras procedentes de otros países, dice, con las mismas garantías, lo dice así, previstas en esta ley, en la Ley de Investigación Biomédica. Las mismas garantías no significa el mismo procedimiento exacto de días o de plazos o de formularios, significa garantías generales de la investigación. Significa que haya habido un consentimiento para la obtención de la muestra que esté acorde con la legislación de ese país y además no sea contrario a la investigación, a lo previsto en nuestro país. Pues, por ejemplo, que por la fuerza no se pueda extraer una muestra a alguien y obligarle a que la transfiera. Nos puede parecer disparatado, pero a lo mejor pues no lo es tanto. Por la fuerza o sin absoluto conocimiento, por ejemplo, vamos a decir. Si en el origen se cumple esto, las muestras pueden ingresar en el biobanco y entonces se integrarán en la política del biobanco con un acuerdo de transferencia. Y el biobanco dispondrá de ellas en los mismos términos que si se hubieran obtenido en España. Al revés, las muestras también se pueden trasladar de un biobanco español a un investigador extranjero. La Ley de Investigación Biomédica no hace distinción. No hace distinción a la hora de la distribución de las muestras. Lo que hay que hacer es cumplir la normativa, la misma. El proyecto que solicite las muestras necesita el dictamen de su comité de ética francés, holandés, el que sea, pero ese dictamen lo tendrá que aportar y tendrá que ser por lo menos entendible por el comité del biobanco, que lo va a dar por bueno. Tendrá que tener unas garantías en el destino, incluyendo unas garantías de protección de datos. Y por eso es tan importante la diferencia entre un país europeo o no europeo, porque en un país europeo la transferencia de datos no se considera transferencia internacional. Es igual mandar el dato a París que mandarlo a Murcia. Igual. Pero cuando hablamos de terceros países hablamos de transferencias internacionales y la cosa se complica mucho, porque hay que demostrar por vías complejas un nivel de protección equiparable al europeo, y esto no es sencillo. Se tienen que garantizar también los derechos de los sujetos, por ejemplo, el derecho de retorno de resultados, aunque ese derecho no exista en el país del investigador. Bueno, como estamos aplicando el régimen del biobanco, ese derecho lo tendrá que garantizar también y lo tendrá que firmar en el acuerdo de transferencia, que será el mismo, probablemente en inglés, pero será en los mismos términos. No hay ningún problema añadido. Quizás el único matiz importante es el dictamen del comité, que a lo mejor no es tan fácilmente validado por un comité español y puede hacer alguna pregunta. Por favor, está en holandés, lo quiero en inglés, o exactamente a qué proyecto se refiere, algo así. Cuando los datos se transfieren, como decía la Unión Europea, no estamos hablando de transferencias internacionales y sin más necesitaremos garantizar la pseudonimización, las garantías en destino, los derechos de los sujetos igual que aquí y, eso sí, identificar las posiciones jurídicas de todos los actores que estén en el circuito de los datos, para saber a quién corresponden las obligaciones y los derechos. También podemos profundizar un poco más en esto, si queréis. En el tema de la pseudonimización, también luego, si hay tiempo, me gustaría comentar una cosa, porque hay una... bueno, ya sabéis que la pseudonimización es una especie, para entendernos, de codificación, donde el receptor no puede identificar al sujeto de los datos porque no tiene acceso, digamos, a la llave del código. El código y los datos identificativos unidos al código se lo queda el que transfiere los datos, y el que los recibe solo ve un código que no tiene ningún significado para él, pero se mantiene la trazabilidad porque tiene un interés, porque probablemente luego habrá que reidentificar por alguna razón, añadir más información, es necesaria la trazabilidad. Los datos pseudonimizados, para el que los recibe, y os lo voy a decir así tal cual, son de carácter personal, en estos supuestos. Pero me gustaría matizar esta afirmación, porque no es pacífica, por ser honesta, ¿no? Para terminar, el último punto. Nos vamos a encontrar ante un panorama muy novedoso. A partir de que se empiece a implementar, se haga efectivo el reglamento sobre el espacio europeo de datos de salud, que se publicó el 5 de marzo del 25, pero ahora os he puesto un esquema de los plazos de implementación. En España se está trabajando en un anteproyecto de ley de salud digital que es necesaria para solucionar muchas de las cuestiones que se plantean en el reglamento. Por ejemplo, va a haber que identificar autoridades nacionales encargadas de desarrollar el reglamento y lo va a tener que diseñar la ley, y nosotros vamos a tener que saber quién está a cargo de todo esto. Eso, entre otras cosas. Ahora mismo se abrió una consulta pública simplemente para, así como una primera llamada a los actores implicados en que demostraran algún interés. Yo he participado en alguna respuesta en el marco de por ejemplo, de Impact Data. Hemos mandado algunas sugerencias que eran importantes para el programa. En dos pinceladas, porque no tengo mucho más tiempo, pero si luego os apetece profundizamos también. En dos pinceladas, el reglamento se entiende en dos bloques. Un bloque que se llama regulación del fin primario de los datos y un bloque que se llama regulación del fin secundario de los datos. No voy a entrar en el fin primario porque se refiere a las historias clínicas con la finalidad de atención clínica, o sea, bueno, diagnóstico, tratamiento, pronóstico, etcétera, siempre en una relación médico-paciente, asistencia sanitaria. Ese es el primer bloque. Fundamentalmente se trata de avanzar en la interoperabilidad. Vamos a dejarlo ahí, en lo que más nos puede interesar para RENACER, porque luego el fin secundario se va a nutrir de estas historias. Si son ya interoperables, si ya tienen criterios de calidad, si los datos están bien estructurados, eso luego va a pasar a investigación también. El fin secundario supone la obligación para todos los tenedores de datos de salud de poner a disposición esos datos con determinados fines, que ahora voy a señalar. Cuando digo los tenedores de datos de salud, me refiero a todos los tenedores de datos de salud, incluyendo los biobancos y sus datos, incluyendo los investigadores y sus bases de datos de sus proyectos de investigación, incluyendo todos los hospitales de Europa y sus historias clínicas. Los ciudadanos tendremos derecho a la autoexclusión, pero no nos van a preguntar, ¿queréis participar? Nos tienen que dar la oportunidad de querer excluirnos, es decir, yo no quiero participar. La autoexclusión es el famoso opt-out, que deben establecer en qué términos se hace en cada país, deben establecerlo los estados. Vamos a ver en qué términos se hace en España. Fundamental es que todos los tenedores se llaman así. En una traducción de data holder se ha traducido por tenedores de datos. Los tenedores de datos van a tener que participar en un catálogo nacional de datos, de manera que se va a centralizar en un gran catálogo nacional todas las bases de datos que hay disponibles. Eso será una obligación vuestra, de los tenedores, aportar vuestros catálogos, siguiendo las indicaciones que se den en el futuro por la Comisión. Este reglamento va a desarrollarse a través de muchos actos de ejecución, que se llama, que son decisiones de la Comisión con expertos, apoyándose por los actores implicados, sobre muchísimas cuestiones técnicas, entre ellas cómo tienen que ser los datos, qué calidad tienen que tener, cómo tienen que ser los catálogos, y aquí no me preguntéis porque no manejo bien este tema. Hay fines prohibidos y fines permitidos. Estos tenedores tienen la obligación de disponibilidad de los datos para unos usuarios, se llaman así los destinatarios. Es posible, dice la normativa, establecer garantías reforzadas para ciertas categorías de datos como los genéticos. Esperemos que esto no lleve a bloqueos. Se regula por primera vez en el nivel europeo el derecho al retorno de los resultados por parte de los sujetos. Y la gestión va a corresponder a lo que se llama un organismo de acceso, que va a haber como mínimo uno por país. Aquí seguro que hay 17, yo creo. Va a haber tiene que haber un coordinador. Bueno, las solicitudes se hacen a través del organismo de acceso. El organismo de acceso las examina, contactará con el tenedor y le obligará a ponerlas a disposición del usuario, ya veremos cómo. En un espacio seguro que hay que crear, unas infraestructuras que hay que generar, y en alguna ocasión también puede ser la propia estructura del tenedor de los datos cuando es un tenedor de confianza, que es una figura que yo creo que puede encajar con el biobanco. Ya se habló en Bilbao en un congreso que tuvimos ayer y antes de ayer sobre biobancos europeos. Hay algunos tenedores que tienen ciertas particularidades, son muy confiables y pueden tener aquí un papel muy importante en el espacio de datos y también tomando decisiones. Y los biobancos responden mucho a esta idea. Importante, esta infraestructura no es excluyente. Convive en paralelo con otros mecanismos que estén establecidos para compartir datos. El biobanco va a poder seguir actuando perfectamente igual. Lo que pasa que también va a tener este circuito. Vamos a ver cómo funciona este circuito. Si no funciona bien, pues ganarán los otros. No sé si decir ganarán o bueno. Y luego otra cosa, esto es un circuito de datos, no de muestras. Va a tener que convivir también, si lo que se manda en un paquete es una muestra, va a haber que hacer un acuerdo entre el biobanco y el destinatario y no un acuerdo a través del organismo de acceso. Bueno, estos son los fines permitidos y me quería centrar en los fines permitidos para solicitar estos datos. Me quería centrar en este, fijaos, que incluye la investigación científica. Específicamente se hace una mención de la generación de servicios y productos, ¿no? Generar servicios y productos, entrenamiento de algoritmos, por ejemplo, para que no quede duda que esto se puede hacer, que no es algo, digamos, meramente tecnológico, sino que incluso aunque haya procedimientos que no generen conocimiento, sino que lo validen, etcétera, también será legítimo el uso. Bien, esto ya lo hemos comentado, así que finalmente esto es un poco, es una diapositiva de la Comisión Europea y es el timeline, que es indicativo además, pero veis que hay un timeline muy importante en 2029, ¿no? Pero bueno, antes de eso tenemos que estar preparados, porque se van a empezar ya, se supone que ya va a estar disponible esta posibilidad de intercambio. Pero esto va a llegar hasta el año 36, cuando hablemos de abrir el espacio a terceros países, por ejemplo. Esto está previsto para el 36. Con esto ya termino, muchas gracias. Así que si queréis comentar algo más, pues estoy a vuestra disposición.
¿Alguien tiene alguna pregunta para Pilar? Perdón. Sí, Carmen.
¿No va? ¿Ahora? Sí. Bueno, he vuelto de Bilbao con mucho que estudiar y un poco emparanoiada, no te voy a mentir. Pero tengo una curiosidad respecto a lo que has dicho de que los datos genéticos en el uso secundario también siguen siendo de carácter personal. Si un donante revoca el consentimiento, ¿qué implicación tiene eso para nosotros? O sea, se supone que habría que eliminar todos sus datos personales, pero, por ejemplo, en el contexto del espacio europeo de datos, si ya hemos volcado esos datos en un repositorio público, entiendo que ya hemos perdido el control respecto a esa parte de los datos, ¿no? Entonces,
vale.
saber...
La revocación del consentimiento tiene efecto, dice la ley dice en general la legislación, la revocación del consentimiento tiene efecto a partir del momento en que se produce y no afecta a los tratamientos legítimos hechos con anterioridad. Además, en particular para la investigación, la ley de investigación biomédica dice que, reconociendo también el esfuerzo de los investigadores y el trabajo que han llevado a cabo con esos datos, dicen que los datos, que las investigaciones desarrolladas con datos hasta el momento del consentimiento seguirán manteniendo esos datos, por ejemplo, si son necesarias para presentar evidencias o algo así. Lo que no se podrá es seguir utilizándolos a partir de ahora en un nuevo proyecto, no se podrá, no. Dices, bueno, ¿y si están en un repositorio? ¿No? Está en un repositorio a lo mejor también para que se compartan, para que se consulten. Bueno, si ya no son identificables, estamos hablando de otra cosa, porque si ya no son datos de carácter personal y se han anonimizado, la revocación del consentimiento no les afecta. Si se ha mantenido la trazabilidad, se pueden hacer dos cosas, o romper la trazabilidad en la revocación, en ese momento, o retirar los datos, sí. O sea, sí, en ese caso, si no se pueden anonimizar, si se tienen archivados en un repositorio para que se sigan utilizando en el futuro y hay una revocación, pues, y es que ese mecanismo además debería estar previsto, ¿sabes? Porque esto debería estar un poco preparado desde el principio. No sé si te he contestado. Sí.
Hola Pilar, yo tenía también otra duda sobre cómo se hace esta compartición cuando tenemos también muestras y datos asociados. Porque no sé si se va a establecer un mecanismo por el que las muestras se comparten por una vía y los datos se tendrían que compartir por otro, o los biobancos podemos hacer una acción conjunta donde se compartan de manera coordinada, por no duplicar también los esfuerzos de investigadores que nos solicitan esta información.
Te refieres al espacio europeo de datos sanitarios, ¿no? Bueno, primero, vosotros podréis seguir gestionando las solicitudes de muestras y de datos por vuestra vía tradicional cuando haya un investigador que se dirija a vosotros directamente. No es excluyente. ¿Qué pasa si hay un investigador que os llega vía organismo de acceso? ¿No? El organismo de acceso os dice, este investigador quiere datos de vuestro biobanco. Lo que dice el reglamento es que vosotros vais a evaluar la solicitud, vais a dar una propuesta de respuesta, pero es el organismo quien da el permiso y quien, digamos, tiene la relación directa con el usuario, con el solicitante. Eso es lo que está ahora en el reglamento. Vamos a tener una ley de salud digital, donde tendremos la oportunidad de que los actores que se van a ver involucrados aquí planteen sus particularidades. Y, por ejemplo, se podría establecer algún mecanismo de... A ver, por el organismo de acceso se va a pasar, o sea, eso es prácticamente seguro. Pero a lo mejor puede haber un mecanismo en que el organismo de acceso supervise, esté informado, pero permita un contacto más directo con el usuario, vía también trasvase de muestras. Pero es que toda la gestión del trasvase de muestras no la vas a trasladar al organismo de acceso, o sea, es que eso jamás lo vas a trasladar. Entonces, lo único que se podría pedir es que hubiera un hilo, además de revisar la solicitud y que el organismo de acceso sea el que le dé a la tecla y le diga si accede a los datos, luego tenga él el contacto con el usuario, el biobanco a lo mejor podría pedir estar involucrado en esa relación o ofrecer por la misma vía al usuario la posibilidad de añadir muestras en un envío, pero eso no está resuelto. Y ahora es una oportunidad de pensar todo el mundo en mecanismos imaginativos que, respetando las normas generales del reglamento, que son muy generales, o sea, sin saltárnoslas, mecanismos imaginativos que hagan más operativo el procedimiento. Dentro de unos años, faltan unos años. O sea, que es el momento de ver dónde están, digamos, yo no diría obstáculos, pero bueno, ver dónde están los posibles puntos de fricción, pero aportar soluciones, claro, posibles, operativas. No sé, eso te puedo contestar.
Hay diferencias de requerimientos para el acceso a muestras y a datos en los países europeos, por ejemplo, a nivel de la necesidad de una aprobación ética.
¿Perdona?
A la necesidad de una aprobación ética,
sí,
que en España es obligatoria para todos los proyectos que usen muestras y datos de origen humano, pero no es así en otros países. En este contexto de compartición internacional de datos, esto ¿se sabe hacia dónde va, la normativa? ¿Cómo se va a gestionar este tipo de cosas?
A ver, hay dos revisiones éticas, revisión a nivel ético en este futuro espacio. Se dice que los organismos de acceso van a tener que revisar los aspectos éticos que yo entiendo que debería ser de la cesión. ¿No? Si es que esto se parece mucho a la estructura de los biobancos, si lo piensas. Y el biobanco no se pone a estudiar el proyecto, porque eso ya viene estudiado, ¿no? Establece si es ético ceder esta muestra a ese proyecto. Bueno, pues lo que dice el reglamento es que esas transferencias tienen que pasar una evaluación ética. Pero también, y esto se tiene que aclarar cómo, porque no se dice quién, le corresponde a los organismos de acceso que se puede apoyar en otras entidades. Se tendrá que aclarar en la ley de salud digital. Pero a mí me recuerda mucho al director científico que se apoya en el comité de ética externo. El comité de ética externo no se mete en evaluar proyectos, solo hacen los comités de ética de los centros que evalúan sus proyectos. Aquí, cuando se solicitan unos datos, se tiene que acreditar que ese proyecto cumple unas ciertas garantías éticas. Y ahí, ahora no recuerdo exactamente los términos, pero ahí, claro, en España aportaremos la evaluación del comité de ética. Digo que no recuerdo exactamente los términos en que se pide esta acreditación ética, pero se pide. Entonces, lo que es muy fácil es presentar una evaluación de un comité de ética acreditado. Y si no, habrá que justificar por qué no o aportar otros mecanismos válidos también.
Sí, yo me
o sea que en ese
refería
sentido sí que se va a unificar.
Me refería a proyectos desarrollados en el exterior de España que solicitan datos a un tenedor de datos español.
Sí, pues este es el mecanismo, sí.
Va a seguir haciendo falta.
Sí. Una evaluación ética del proyecto cuando se hace la solicitud. Yo creo que no se dice exactamente el dictamen de un comité, ahora lo voy a mirar, porque no me acuerdo exactamente.
Vamos a Pilar y seguimos con las dudas que hay luego. Muchas gracias,
Muy bien,
Pilar.
gracias.
Bueno, nuestro último ponente de hoy es Sergi Benavente. Sergi es radioterapeuta, está trabajando en el Hospital Vall d'Hebron, oncólogo radioterapeuta. Está especializado en cabeza y cuello y también en radiocirugía y enfermedad metastásica. Tiene una larga carrera clínica y también investigadora, estudiando los mecanismos de resistencia a las terapias en cáncer de cabeza y cuello y metástasis cerebrales. Participa en muchos consorcios europeos, en el ARFOS, en el E2Radiate, en el Oligocare y en el RICARE, y también en el Systemic Therapy Focus Group de la Sociedad Europea de Oncología Radioterápica. Nos va a hablar de los retos actuales en la irradiación de metástasis cerebrales. Y muchísimas gracias, Sergi. Es miembro también de nuestro comité de dirección, no se me olvide decirlo. Así que muchísimas gracias.
Thanks for the invitation. Thanks to the organizers. So my talk is switching gears a little bit. It's what can radiation offer in the personalized medicine context. So first of all, what could be the right local treatment, if any, for brain metastasis? We all know the benefits and the inconvenience of surgery and radiation. We know that surgery benefits a subgroup of patients and that radiosurgery is less toxic. And from that, the general agreement is that radiosurgery is a standard treatment, either in the postoperative setting or in the radical setting for a given patient. We also know that talking about results implies a lot of heterogeneity, not just biological heterogeneity, but clinical heterogeneity from the patient, the treatment stage, and in this slide, just stratifying by the performance status of the patient, you can see that any treatment shows significant differences. So that's really important. What else? What we have improved? At our center, we have a brain metastasis dedicated tumor board. We have been doing that for probably about 15 years now. It's a really good option. Radiosurgery has great acceptance. We can reach more patients this way. And finally, technological improvements offer more options. Coming back to personalized medicine, this is a momentum for brain metastasis. It's a really unmet need. Poor prognosis for many patients, many new pharma options, targeted therapies, immunotherapies, new treatments, and finally, we need to think about the patients. So with that in mind, I'll be showing you some examples that summarize some of the information that I'll be talking about. So this is, for example, a young woman with relapsed melanoma, with a mutant BRAF melanoma. She had polymetastatic disease and one brain met, which we treated with radiosurgery. You can see it here. And this is the control right now. This is about a year ago. This is clean, no imaging alterations or anything, but she has a new lesion. She has switched treatment, and we will be treating her next week. So we can perform more treatments per patient. That is an added value. In this context, multidisciplinarity is paramount of this, in these situations. But we need to do better to standardize the patient journey, let's say, and to try to include CNS endpoints if it's a CNS-directed activity or not. There are many difficult things to be sure or to, for example, symptomatic or asymptomatic, if it's an active or non-active lesion. All of that is complex. The management can be very complex. For example, in this case, 2018, a 56-year woman with an exon 20 EGFR mutated, locally advanced non-small cell lung cancer that with synchronous brain metastasis, she had these these 6 lesions here. She received immunotherapy. We did radiosurgery for those lesions. Next chapter, she developed a new lesion, this one here. We did radiosurgery. This lesion here was never controlled. There was no surgical option. So we performed a second radiosurgery for that lesion. And the patient switched treatments. And at some point, she developed all these other new lesions, 8 new lesions. We could certainly treat those with radiosurgery, but this lesion here was still growing, and the patient was oligosymptomatic, so we decided to not perform whole brain radiation. She kept going on different targeted therapies and chemotherapies, and at the end, quite some time later, she died of neurological death. So this is the complexity that we are usually facing. CNF active systemic therapies. So what is response? How good is good? Long-term situation. So it's very difficult to find a consensus on that. Some people are just proposing, simple options to go, but I think that the real point is to think what is adequate or not adequate as a response, and what happens with the long-term survivors, not just in quality of life, but in control of the lesions. One aspect that I'm very much involved is with regard to safe combinations. We have many different options. Globally, let's say that it's assumed that immunotherapy combination with radiosurgery or radiotherapy is safe. There are some concerns with ipilimumab and melanoma, but not sure at some point. And there are some concerns with anti-angiogenic and with multi-target TKIs. I will be showing in this part over here because many of these are European efforts, and many of them are trying to group a broad population. So new treatments. This was a patient with triple-negative breast cancer that at some point developed this 3 by 3 centimeter brainstem lesion that we treated with radiosurgery and the systemic treatment in this case was pembrolizumab. She did very well. The residual lesion was almost fairly visible, but at some point she presented with leptomeningeal disease. At this point, she started an ADCs, sacituzumab govitecan. She regained functionality. She had an ECOG3, and then she went back to an ECOG1. But at the end, the leptomeningeal disease progressed, and that was the cause of death. But no safety concerns at this point. So that's an additional thing to bear in mind. Leptomeningeal disease, it's, well, certainly our contribution is mainly palliative. Just two comments on that. Maybe some of you have seen that in the last year there was a paper, a small clinical trial showing that proton chronospinal irradiation offered better results. Certainly it does, but it's not probably the context. But I mean, creativity is out there, just to keep in mind. And then another situation that is fairly more common, which is the postoperative nodular leptomeningeal disease. In this case, the prognosis is better, and sometimes we can offer more aggressive treatments, like, for example, in this situation. This is a 38-year woman with HER2-positive breast cancer, locally advanced, optimal response after treatment, that a little bit after treatment completion, she presented with these two confluent brain mets that were taken out, and then we performed radiosurgery of the surgical cavity, January 23. July 24th, the signal changed. In the MRI, we thought it was meningeal disease. We confirmed that with PET, with an amino acid PET, and then she underwent a second course of radiosurgery to the surgical cavity. This is today. Last week, I saw the patient, and there is no perfusion here, so we are assuming that everything is good so far. Finally, the most important and global aspect for me is neuroprotection. Europe, I mean, the European Society of Radiotherapy has taken this very seriously, and this is a strong pillar right now to take into consideration. So, I mean, we know radiation, radionecrosis, adverse radiation effects, which means that something's strange in the MRI, but the patient has no symptoms. That's completely different. I mean, radionecrosis may be up to 20% when it should be, in fact, below 10%. Well, that's the numbers. It's difficult to identify it. We have some advanced MRI options, the amino acid PETs. It's also difficult to quantify the intracranial response. And finally, two simple things is that neuroprotection, whenever there's the chance, radiosurgery is not the problem of the number of metastases is going to be the best neuroprotective option. Otherwise, there are several different modified whole brain radiation options or whole brain radiation. It just needs to be adapted to the situation of the patient. And finally, something that, I mean, I thought I would put it here because we're talking usually about quality of life, but now there's something called quality of care. So we may need to get into that just to make sure that we feel that the patient has received the best attention, but that the patient is feeling it the same way, has no different opinion on that. And with that, I thank you for your attention.
So any question in the audience?
Ángel Rodríguez, I'm neurosurgeon. Congratulations for your presentation and those beautiful cases with a good, very difficult surgical cases and a good response. One of my questions is, what is your opinion about the intraoperative radiotherapy that I know in Barcelona, in Belviche, they are starting to use that. And the other question is, what do you think about after surgery of a brain metastasis, not to go to the local radiotherapy, instead of delay the radiotherapy and use the immunotherapy, for example, as the oncologist, the medical oncologist that showed before with his presentation. And what do you think? What is your opinion?
With regard to intraoperative radiotherapy for brain metastasis, I mean, it's an option. It's been there for many time, I'd say. One of the main issues is that, I mean, availability is not necessarily the worst part, but the most important thing is that now there's an important debate about the efficacy, not for this situation, but of intraoperative radiotherapy. I mean, technology evolves, and I'm not really sure that at this point this is, as competitive as some other options. With that said, there are many patients, many situations, salvages. I mean, there are many needs for the patient. So probably it's there for some patients, and that could benefit from that. That would be my take. With regard to the second question, if we can defer or avoid radiation after surgery, I agree. I mean, I agree. I do not need to agree. That's data. So, I mean, there's a benefit with pembrolizumab, but as I showed in one of the first slides, there's a lot of work to do, and probably they are there. We don't know who they are, but there are some long or very long survivors, and that's one. And second, it's that it's like there are low risk and high risk, let's say, for brain metastasis. So I think we would have to include some more stratification into that, and I would not avoid or defer radiation. I mean, not sure how strong the data for pembrolizumab is, but as you know, I mean, the potential recurrence after radiation with no additional treatment still requires some additional effort. So that's what I would do. I would keep both, if possible. That's what we do if the context is right.
So, Sergi, thank you very much. How frequent is to do this? I was not aware of radiosurgery for new lesions. So, after a patient has been irradiated receive multiple rounds of stereotactic for new lesions. So, this distal relapse. And what are the limitations? I imagine that it depends where is the new lesion. If it's more close or far from the original lesion, that might have some limitations because of potential effects.
So the big change, let's say, is that you can treat, I mean, the difference or the handicap of radiosurgery versus whole brain radiation, let's say, would be that there are more intracranial relapses with radiosurgery. Okay. The good point is that we can do as if it was the first time, because usually a new lesion is not a problem. Usually intracranial relapse is somewhere far from the original lesion. But even if it was close, we can still manage to do that. That's one thing. The other thing is that, and the work case is a little bit mixed with all these situations, is that we cannot solve the initial problem. We have one brain metastasis that is not doing good. So it keeps going. So that's a different situation. We can do re-irradiation of that. Toxicity raises somehow a little bit, and probably we can. That was the point with that patient. We would not do a third procedure for that. And then the other thing is that it's not exactly, but I mean the number of lesions. At some point, when this case I presented with 6 lesions, at that time we had to go one by one. We did that a little bit different, but that's the point. At this moment, we can do that all them in 30 minutes, which is the same time that requires treating one. So there are a lot of conditions. There are a lot of situations. Certainly, if it's in a location that, let's say, in an eloquent location, it would be before, the first option, if possible, would be focal radiation somehow. If that's not an option, well, we would have to discuss between, and then the point is that a combination and concomitant treatments is probably the best option at this point if we can find an equilibrium, a balance of the situation for the patient.
Thank you. It's very complex. Yes.
Okay, so thank you very much, Sergi.
Thanks.
All right. Go ahead. So we have completed the sessions, the invariant talks. Vamos a pasar a castellano a lo mejor. Tenemos siempre al final una ronda de preguntas de cualquier cosa que tenga que ver con la red, problemas que no hayamos tratado, sugerencias que podáis tener. Esto es una asamblea general, no es un congreso, entonces cualquier input que tengáis, por supuesto, se puede hacer. Obviamente no estamos todos los que, ni mucho menos todos los que estamos en RENACER, pero ahora se abre una mesa de preguntas, sugerencias que podáis hacer, etcétera, ¿no? Pensad si tenéis alguna pregunta. Y luego el otro punto... sí, Ángel.
Es referente al test neurocognitivo. Nosotros en Castilla-La Mancha pues, es una población muy rural, ¿sabes? Entonces a veces es difícil el que el paciente acepte, o sea, al principio acepta, ¿sabes? Lo que pasa es que luego cuando empieza a ver que le mandas el correo electrónico, tienen que mandar un correo electrónico, entonces ahí el paciente se echa para atrás. Entonces mi pregunta, o sea, es ¿qué podríamos hacer para esos pacientes? Porque nosotros tenemos un ordenador en el servicio que podíamos dejarles mientras están ingresados, pues a lo mejor facilitar un poco para ese tipo de pacientes, ¿sabes? Que sea más fácil el que a lo mejor simplemente clicando un enlace, pues ellos entren y puedan hacer el test. No sé si habéis pensado en alguna cosa así.
O sea, ahora mismo es así. O sea, ahora mismo, claro, nosotros lo que necesitamos es un correo electrónico.
Claro, pero...
o sea, dices que esté instalado en un ordenador y vaya allí y lo haga.
Sí.
A ver, yo creo que lo que estamos viendo que funciona mejor es que haya una persona que esté al cargo de hacer esto y pueda dedicarle tiempo a estar con ese paciente para supervisar que hace el test. Entonces esto, por ejemplo, en el Hospital 12 de Octubre lo está haciendo Blanca Navarro y ahora en la Princesa Alicia González, que es un clínico. Y esto estamos viendo que es la manera de que funcione. Lo estamos testando todavía. Está clarísimo que esto es un problema, no solo para vosotros, sino para todo el mundo. O sea, está claro que la gente, los pacientes no entran, cuando entran no completan y llevamos meses intentándolo y llevamos un paciente que ha completado. Sí. Entonces, por eso estamos pensando también otro tipo de estrategias que puedan ser implantables en varios hospitales, porque el problema es que no todos los hospitales pueden tener una persona, un psicólogo clínico, una persona especializada en atender a un paciente, claro. Entonces, es un equilibrio que, o sea, no tenemos una fórmula definida para hacerlo. Ahora estamos registrando que hay un problema y como lo habíamos pensado no se puede hacer.
Sí, entonces, pero en cualquier caso, si vosotros tenéis opción de un ordenador y un sitio donde el paciente puede estar, quizás sí podríamos hacer un circuito como para enviaros a un correo que pudiera estar en ese ordenador, digamos, a lo mejor no de nadie en particular, pero y que desde ahí pudiera acceder al enlace. Esto creo que no habría problema en hacerlo. O sea, facilitáis la infraestructura.
Y cualquier iniciativa hay que probarla, porque eso es más sostenible que pensar que va a haber una persona en cada sitio que va a estar encargado. O sea, si es cuestión de tener en cada sitio un ordenador donde esté el programa y esto facilita que las cosas se hagan, pues tendremos que conseguir financiación para tener ordenadores específicos en cada centro y eso es totalmente factible a priori, o mucho más factible que tener una persona dedicada. Entonces, si vosotros podéis hacer esa prueba, yo creo que hay que intentarlo. Yo creo que sí. ¿Alguna cosa más que queréis hablar?
Bueno, yo quería traer una reflexión general sobre cómo mejorar la participación de todos los perfiles que están formando parte de la red. Bueno, ya sabéis que María Jesús y Manuel le ponen mucho cuidado a tratar determinados, variedad de temas que puedan ser de interés para todos los perfiles recogidos dentro de RENACER, y era un poco pues traer esa reflexión de cómo podemos abordar estos encuentros para que sean pues más interesantes para los perfiles médicos que participáis, cómo también traer información relevante para los investigadores, incluso cómo llevar esto hacia los pacientes, que también es algo que estamos persiguiendo, cómo comunicar mejor los resultados que traemos. Entonces, bueno, pues aprovecho para traer esta reflexión a este encuentro, a ver si surgen ideas interesantes.
Sí, desde luego es el interés de Manuel y mío de siempre, favorecer el que la información entre todos se comparta y las iniciativas se compartan. Y las asambleas al final son la herramienta que tenemos más a la mano para encontrarnos y para hablar y cara a cara de todos los problemas. Sí que invitar a la participación de todos los centros, que además es una obligación que tenemos, porque todos respetamos los estatutos y en los estatutos la participación en las asambleas es algo que está recogido como algo obligatorio para los centros. Que no se trata de obligar a nadie, se trata de favorecer que vosotros os sintáis pues de alguna manera con ganas de venir, porque os vamos a aportar información relevante para vosotros y al revés, que todos trabajemos en equipo. Esto es un equipo, al final, todos tenemos que participar, ¿no?
Una de las cosas que habíamos pensado, que también compartirlo, aunque no... O sea, yo creo que el perfil fundamental que falta son los clínicos y es una realidad. Hay más dificultades que en otras profesiones para poder acudir a un tipo de evento como este. Entonces, una de las opciones que estamos barajando es asociarnos, ya sea siempre a una sociedad médica o clínica, para hacer esta asamblea digamos, cuando ocurra este congreso anual donde soléis ir. Obviamente ahí tendremos la dificultad de que no todos los clínicos, o sea, dependiendo de la especialidad, pues vais a un congreso y no podemos hacer 4 asambleas diferentes, ¿no? Entonces, esto, quiero decir, hay que hacer algo de brainstorming, pero claramente por lo menos la única experiencia que hemos hecho, sí que hemos visto que obviamente pues, había más asistencia, y esto es una opción para ayudar a que podáis acudir los clínicos. No sé, si tenéis ideas, por supuesto, por favor, compartidlas con nosotros. Lo que mencionaba María G, y ahora te dejo hablar sobre esto, pero uno de los problemas que ahora estamos teniendo por primera vez este año es que al ser una asamblea, al ser una red, tenemos que votar cosas. No podemos tomar decisiones. María G, un día se le ocurre algo, no puede tomar una decisión. Esto hay que votarlo y tiene que hacerse, el momento de hacerlo es la Asamblea General. O sea, solo podemos cambiar los estatutos de RENACER en Asamblea General como la que tenemos ahora. ¿Cuál es el problema? Pues que obviamente no todo el mundo puede venir, totalmente entendible. Hemos dado incluso la opción de delegar voto por correo, pero tampoco ha funcionado. ¿Cuál es la consecuencia? Que no podemos aprobar una modificación de estatutos que creemos que es buena para la red. Esto es un experimento piloto, no nos vamos a morir mañana si esto no se cambia hoy, porque no es una cosa, digamos, crucial, pero quizás puede que haya cosas que sean más relevantes y que sí que haya que ejercer esa capacidad ejecutiva, ¿no? Entonces, esto yo creo que ha sido un ejemplo para darnos cuenta de que tenemos que encontrar una manera de que podamos hacer esas votaciones, que los miembros de RENACER puedan ejercer su derecho a voto y que vean la importancia de hacerlo, porque esto, desde luego, es lo que nos permite seguir trabajando en una dirección o no. Entonces, bueno, tenemos que pensar cómo hacerlo, está claro.
Sí, un poco volver a la idea esta de que realmente esta red es una red participativa y colaborativa. Desde el minuto cero así se diseñó y así debería funcionar. Entonces, sí que me gustaría que los clínicos os sintierais involucrados también como parte del equipo. Nosotros os sentimos parte del equipo. Y bueno, pues me gustaría poder llegar.
Sería una opción, por ejemplo, los que estáis aquí, que sois clínicos, ¿sería más fácil para vosotros que esta asamblea estuviera, o creéis que vuestros compañeros que no han podido venir verían más factible poder ir a la asamblea si estuviera asociada al congreso en el que vais anualmente? Sería una opción, por ejemplo, hacer esto. Esto lo hemos probado con Heino, por ejemplo, y creemos que ha funcionado. No sé si alguien puede tener alguna opinión al respecto.
Sí, hombre, a mí me ha sorprendido, porque yo creo que otras veces había más clínicos. O sea, yo creo que si cada hospital tiene un IP, ¿no?, de cada hospital, lo lógico sería un poquito decirles a los que están como representantes en cada hospital que, bueno, que tienen que hacer el esfuerzo. Y lo de que comentas de una sociedad, la Sociedad Española de Neurocirugía tiene un grupo de trabajo de neurooncología que se reúne todos los años en marzo y ahí, pues, desde luego yo creo que sí, pero quizá a lo mejor es mejor si se va a asociar RENACER a una sociedad científica, pues una sociedad a lo mejor quizá más potente, como puede ser la Sociedad de Oncología Médica, por ejemplo, que es el Heino, forma parte también ¿no? Es un grupo, digamos, una sección del este. En lo que respecta a los neurocirujanos, yo creo que sí, que en la reunión del grupo de trabajo de neurooncología, yo creo que allí sí que habría más afluencia, porque muchos de los IPs de los hospitales están metidos en ese grupo de trabajo.
Luego también es verdad que lo que implementamos, yo creo que fue el año pasado, en vez de tener una asamblea general en persona, hacemos una asamblea general virtual también, donde nos estamos planteando pues que ahí se ejerce el voto para cambiar estatutos, porque está claro que en este formato es más complicado. O sea, eso también está ahí implementado y yo creo que eso funciona bien, pero yo creo que tenemos que empezar a estudiar esta opción. Yo personalmente coincido contigo en el sentido de que, bueno, pues si hay grupos establecidos en diferentes hospitales y sabiendo con una prioridad de un año que esto va a ocurrir, lo hacemos todos los años, debería ser posible encontrar esta situación, ¿no? Pero bueno, la realidad es la que es y yo creo que hay que afrontarla. Entonces, bueno, sí, Ernest.
Yo hablo un poco desde fuera porque como no me he incorporado todavía, pero sí que es cierto, le comentaba antes a Manuel que el hecho que sea por la mañana a muchos clínicos se puede suponer una dificultad. Y después también, justamente venimos esta semana del Congreso Europeo de Oncología, que de cara a otras asambleas intentar evitar que haya cercanía, porque a veces esto supone la vuelta al hospital, mucha carga asistencial, son días siempre complicados para neurocirugía, en radioterapia, en oncología, para todos siempre nos pasa cuando venimos, igual evitar que o lo haces pegado a un congreso español, como decías, y yo estoy de acuerdo con el compañero que SEOM es una buena opción porque como es una iniciativa pan-tumor, que no es a pulmón o mama, sino que es más general, o yo creo que sería ideal GEINO o SEOM. Y SEOM, siempre las fechas se saben con mucha antelación. Y si no es pegado a un congreso, evitar cercanía a congresos clínicos, porque sobre todo cuando hay ESMO, ASCO, arrastra mucha gente y siempre son semanas complicadas.
Gracias. ¿Alguna cosa más? Pues si os parece, nada, agradeceros mucho que hayáis venido y hayáis hecho el esfuerzo. Y realmente, aunque no estemos todos aquí, los resultados están ahí y yo creo que la red va fenomenal. We have the feedback from Nuria that she's very, interested in the network, that how we are doing, and yo creo que ese es un punto importante, ¿no? El reconocimiento que está teniendo RENACER está ahí y estamos siendo, yo creo, bastante productivos y eso es gracias a todos vosotros, los que estáis aquí y los que no. Así que nada, ahora gracias también a la Fundación Ramón Areces que nos ha brindado un año más este maravilloso auditorio y que ahora muy gentilmente nos va a invitar también a un cóctel, a un vino. Así que nada, daros las gracias y ahora hablamos fuera, si os parece. Sí.
Nos importa que nos hagamos una foto de grupo para que quede el recuerdo de estos encuentros y luego ya vamos al cóctel. Gracias.
Muchas gracias.